Hi everybody, good evening. So we'll just give everybody another minute there before we start. It's not quite 6 o'clock yet, so we'll give everybody another minute. You might just give me a quick hello or a quick thumbs up if you can see the screen, okay, in terms of the notes, and you can hear me and see me as well. Just give me a quick hello or just give me a quick thumbs up. So I know we've got our usual premium members here, so welcome again to you all. But I also know that this is also a week that we're putting free to students. Remember, I'm doing 3 weeks of biology blitz, and this is the first one. So the first one was voted to be free, so we decided we'll give this one free to everybody around the country. But then the other ones we're going to do the other 2 weeks. It's just for our premium members. All right. Good, everybody. Good to see everybody. I recognize a lot of new names and I recognize some old names, so well done. We'll just give everybody another 30 seconds to a minute just before we kick off. I'm good, Kitlin. I'm good. I'm doing well. I was teaching this morning. Who was here this morning? Was anybody here this morning when we did the class from 9 o'clock to 10 o'clock on DNA and RNA? Was anybody here this morning, the second half of that chapter? We got most of it done. You might just give me a heads up there if anybody was there for that one as well. So if you were, that means you'll be here. You'll have seen me 3 hours today. How are you, Carl? Good. All right. So just give everybody another second. If you're new and this is your first time watching this, don't worry. A lot of people are there this morning. Fair play to you. My goodness me. Well done. All right. Oh my God. There's a lot of people there this morning. So well done to you all. Okay, I think we're a minute past 6. So look, hello everybody, and see anybody new as well, welcome. Now, this is the first of our biology blitz. So we're going to do 10 hours of revision this week, and we're going to do 10 hours of revision on the May the 10th, and we're going to do another 10 hours on the May the 24th. So that's 30 hours of revision. Don't forget though, every other week between now and then, I'm also going to be doing Sundays, Tuesdays and Thursdays. But this is our first of our biology blitz. Now, the reason why I wanted to do this is because this is now, things are getting real. We're getting close to the Leaving Cert. And I want to cover, like I see people out there, I want to be honest with you, I see people out there, some people doing social media say, I'm going to do 3 chapters in an hour and a half. There's no way you can do 3 chapters in an hour and a half teaching, all right? There's no way. So I like to do things right. So what I'm saying is I'm doing 10 hours on, all right, these 7 chapters. So let me just show you here. This is the plan this week. We are doing these 7 chapters this week, all right? And I'll tell you which ones we're going to do which day. And what that means is we are going to be studying 30 to 40% of your Leaving Cert. All right, it's actually was worth a little bit more than 40% last year. Okay, so that's what we're doing. So what we're doing is we're going to do enzymes today. We're doing respiration tomorrow. We're doing cell division on Tuesday. We're doing photosynthesis on Wednesday. And these 3 short chapters, cell structure, cell diversity, and osmosis diffusion, we're going to do them on Thursday. All right, and that'll be 30 to 40% of your Leaving Cert. Now, just to point out, all right, people might say, well, okay, what time? Is it the same time every day? Not quite, because I do have 5th years in teaching on Tuesdays as well. So the times are, I'll just show you the times here. So for those that are on the platform, and I'll put them, for anybody that's here for free, because I know some people are here for free. All right, I'll show you where everything is now for everybody. But look at this here. I want to show you this first of all, right? So the first thing, and then we're going to get going. I say we have 2 big hours in front of us. But look at this here. So if you click into my H1 biology platform, and you will see this here. All right, so everybody, we're using our digital notes. A lot of people have the hard copy. All right, so there's the notes on the whole course. But let me show you this here. There's the question bank that we're going to be using today, and the question bank solutions that we're going to be using today as well. But I want to show you this. See this here? Click on this, the Biology Blitz schedule. When you click on this, here is the schedule for this week. Today it's 6 o'clock. Tomorrow it's 7:30. Tuesday it's 8:15. Wednesday and Thursday it's 7:30. So they're the starting times, and we're pretty much going to do 2 hours each day. So that's going to be 30 to 40% of our paper over the next 5 days. That's the first thing. So that's on the platform, but I'll put that up in the news feed as well to keep reminding you, okay? And don't forget the other 2 weeks then. Look at the other 2 weeks. Oh, Look at the other 2 weeks then as well. You'll see them as well. So May the 10th and May the 24th. So much to look forward to there. But those ones are not for free. The other ones are unfortunately are just for, and not unfortunately, but I can't give everything away for free. But this is for the people that are members of the platform. Now, the second thing is this, right? If you want, for most of you that are home, you have my H1 biology notes, right? You have them there. But let's say you want maybe you're a member just for free today and you want to see the notes that we're using. So the notes that we're using are on the platform, and I'll put these up every week. So there's your enzyme notes. See the link there? Just click on that and you can open that there. Now, I've also made this, all right, free. Do you know your biology question bank? And what I've done there is a lot of people, I've printed out 1,500 of them, or 2,000 of them, I think, those H1 question bank booklets. And all of them have been handed out at this point. So a lot of people say, I didn't get one, and they're on the platform. So I said to myself, I let people download it as a PDF. So if you click on that there now, you can download the entire question bank as a PDF. And if you want to annotate it then on your iPad or whatever, you have it there. And I'll just say this one last time before we get going today, everybody. And I know everybody that sees me regularly, I'm always going on about this. And I'm going to say this one more time. If you look at this here in my platform, all right, if you know the questions, sorry, do you see the question bank there? If you know the questions in that question bank from start to finish, you're going to get that H1, all right? I'll just say it one more time. I taught 350 students in a certain day school last year and the year before, another 350, and 350 years before that. And last year and the year before, between 57 to 61% of my students got a H1. The reason why, they work hard, but they know all the questions in that question bank. And if you're struggling, there's the solutions for people on the platform. There's the solutions to it as well. Okay, brilliant. Now, just to remind everybody one last time, for all the members that are full-time members, do not forget all those videos on all the course are there, all right? And any video that's not there, like genetic crosses, I have to finish that one off, and plant reproduction, they're there, but they're there live from the Sunday masterclasses, for example. So my point is, it's all there. Just got to use it. All that on-demand material, and the feedback I'm getting from the on-demand material, where you can just click in any video and watch it on that part of the chapter, and then click on the exam question video based on that content. So many people are loving it. So let's get going. As I said today, you know what the plan is. We're doing 30 to 40% of our Leaving Cert this week. We're going to start off with enzymes, and just to repeat one more time, don't worry if you're a member or a free today and it's just you're here for just a week, I'm going to put up the notes for you just as individual chapters there so you can see them then as a PDF. Okay? Now, let's get going, everybody. As I said, look what the plan is. Look, and see that there? The 7 chapters we're doing, 30 to 40% of your Leaving Cert. And one of the reasons why, because do you see those 7 chapters? And I want to say this again. Do you see question 8? Question 8 has to be on those 8 experiments. And that's 7.5% of your paper. Question 9 has to be on those 7 experiments. Here, another 7.5% of your paper. The reason why, and question 10 has to be on those 7 experiments. The reason why I'm pointing that out today is because, do you see the 7 chapters we're doing this week? See them 7 chapters that we're doing this week? Those 7 chapters have the following. All of these experiments in them. All of these experiments are from those 7 chapters, but also, see here? Also these 2. So this is why these 7 chapters are worth so much, because you're guaranteed one experiment question on question 9, but question 8 could also be based on the experiments from unit 2. So that's why I want to make sure we know this. And I know I've been here since Christmas. We've done some of these chapters now. This is like the 3rd or 4th time I'm doing enzymes. And I've done respiration numerous times. But listen, the more times you see it, the less study you have to do, because you're going to be so well in terms of understanding it. You'll know it so well. So then, because you've understood it so well, it's going to be so much easier to study. Let's get going, everybody. So I want you to open up your, if you're using my H1 exam focus notes, open up page 116, or you might just want to use the ones on the platform on the news feed. It's the same notes. Don't worry, it's the same notes. But most people that are on the platform here, you obviously know you have all the notes on the entire course. So I'm working from them. Okay? Aaron asked me this. Aaron, all of these classes are recorded. Now, I don't know, admin, if you're free here, If you're as a free member, admin might just confirm this. Admin, do free members get to see the recordings on the platform? I don't know. Admin might confirm that. Admin is the person in charge of the live chat there. So they might, geez, admin's ahead of me. All live classes are recorded and made available on the platform for you to watch. But admin, can you confirm, can the people that are here for free for this 1 week, can they watch them? I don't think they can, but the admin might just confirm that. Okay, everybody. So, Aoife, yes, we're just doing enzymes today. Aoife, I like to do things inside out, because I want to make sure everything's covered. So we're doing enzymes today for 2 hours, respiration for 2 hours tomorrow, cell division for 2 hours on Tuesday, photosynthesis for 2 hours on Thursday, and then the other 3 chapters, cell structure, cell diversity, and osmosis diffusion for 2 hours on Thursday. That's it. We'll get them all done. Emma or Aoife, if I try to do any more than enzymes today, I won't do enzymes properly. I want to do all of enzymes from start to finish. Let's get going, everybody. So on page 117, this chapter, as you can see, everybody, look, this chapter comes up so often, pretty much every year. Last year it was worth 6%. The year before that it was worth 13%. The year before that is 8%. The year before that roughly 13%. You get the idea. 7.5%. You get the idea. This chapter is worth a lot. Okay, so let's get going. The first thing we need to know, everybody, is what is an enzyme. So we're going to go through certain parts of the chapter, and then we'll look at some exam questions from our H1 question bank booklet. Let's have a look. So what's an enzyme? An enzyme is a biological catalyst. You got to use them words. Biological catalyst. Now what that means, biological catalyst means it's a biological because it's a protein, and a catalyst because it speeds up the chemical reaction, but it's not used up in the reaction itself. So an enzyme is a biological catalyst. Okay? Now, if we then go down a little bit further, if you then look at this here, you will then see what is the chemical nature of an enzyme. What's the chemical nature of an enzyme? That was asked in 2011 or 2012? A lot of people struggled with that because they didn't understand what was meant by chemical nature. An enzyme is a protein. Another way they asked that question is this: what biomolecule are enzymes made from? Enzymes are made from proteins. Enzymes are made from proteins. Okay, now, this was asked in 2021. Because an enzyme is a protein, where are enzymes made in a cell? Oh my God. So many people couldn't get that. Now, a lot of people knew ribosomes make proteins. A lot of people knew enzymes are proteins. But so many people didn't link the two things together. So, enzymes are proteins. Proteins are made in the ribosome. So therefore, enzymes, which are proteins, are made in the ribosome. And you'll see that there. Now, if anybody was at my DNA and RNA class this morning, they'll even know more about it, because a gene is a section of DNA that has got the genetic instructions to make the protein. So our DNA code ensures that the ribosome joins the right amino acids together. But that's for a different day. We saw that this morning. Now, if you then look at this here, everybody, in 2023, they asked you to name the 3 products that are from this reaction. Now, this is also in human digestion. So this is the link between the 2 chapters. So if you look at this here, George says, I heard enzymes won't come up this year. Is that, George, just ignore that nonsense, George. Nobody knows what's coming up. Enzymes comes up nearly every year. And because, George, there's so many options in the paper now, the paper's got so many extra questions. The majority of the course is asked now. Look, question 16, George, is A, B, C, and D. There's 4 chapters usually in question 16. Question 17 is A, B, C, and D. There's another 4. So question 16 and 17, there's probably 8 different chapters if it's going by the last few years. So, like, that's just two questions with 8 chapters usually. So my point is, there's a lot of extra questions, and some of those questions have extra parts to choose from. So, George, my answer is, I wouldn't bet against enzymes. It comes up pretty much every year, okay? Now, if you look back to what we're saying there, George, so you need to know, all right, lipase, you need to know this for human digestion as well. Lipase breaks down lipids, which is a substrate, and lipase is the enzyme. ASE is always enzymes, yeah? So lipase breaks down lipids into fatty acids and glycerol. Okay, amylase breaks down starch. Amylase is the enzyme. Breaks down starch to substrate. Amylase breaks down starch into maltose. Protease breaks down the substrate proteins into amino acids. So if you named those 3 products, you got 9 marks, 2.5% of your leaving cert. So make sure you know the 3 products of those reactions. And to be honest with you, you should know them anyway, because those are also in the human digestion chapter. You need to know that for human digestion. as well. Okay, so if we then go back here, all right, so admin just confirmed their all the on-demand and the recordings are only available to premium members, so members that are paid, unfortunately. But for those that are here today, just come along to the live classes that are attending the classes for free today, and you'll be able to see them live, okay, over the next 5 days. Now, let's have a look at that. So the next thing is facts on enzymes. You need to know the enzyme shape. This has been asked a few times. What's the shape of an enzyme? They're folded. Enzymes are folded in shape, okay? All right, now, if you then look at the word specific, what do we mean by the word specific there? The word specific means, like, look here, amylase, see amylase here, okay? I'll just rub this out and tidy this up a little bit. So let me rub this out here. So if you look at amylase is an enzyme that will only react with starch, one particular substitute, that's it. And for that reason, we say enzymes are said to be specific. They only react with, highlight the words one particular there, everybody. They only react with one particular substrate. If you come along and say an enzyme reacts with a substrate, no, you're going to get 0 marks for the word specific. It reacts with one particular substrate. All right, let's go down a little bit further. What 2 factors affect the enzyme's rate of reaction? pH and temperature. Now, if they ask you, how does pH and temperature alter the rate of the reaction? Because pH and temperature affects the shape of an enzyme. And if the temperature and the pH is not suitable, that enzyme shape, which we call the active site of the enzyme, see, highlight the word active site. We'll see this now later on in the chapter as well. The active site is the part of the enzyme that changes shape to suit the substrate. Okay? So, if the temperature and the pH is not suitable, then that active site will be altered, and therefore the reaction will be slowed down, maybe even stop. Now, let me just ask you now to turn over the next page. These are all things that have come up in the past. Saoirse just asked the question, since you're not tight for time for the exam, how many exam questions should I do? Saoirse, what I'm going to say to you, anything about the exam paper, I'll talk about at different stages today, but to answer your question very quickly, Saoirse, most people will do extra questions. It just depends how much time you have, but most people will generally do 1 or 2 extra questions. If you look at this, so Katie, it's from your H1 biology notes. Katie, this is on page 118 from your H1 digital copy book. So most people have the hard copy. And if you're here today and you don't have the notes, don't worry. These notes are on the platform, okay, on the newsfeed, so you can download the individual chapter. Okay, so if we then look at this, everybody, so let's go over the next page. Let's get all these little things come up so much in this chapter. So everything in this chapter has nearly always been asked. So there's not much in this chapter that has not been asked. So let's have a look. So highlight the definition of a substrate. This definition hasn't come up, I think, since 2011, 2012. It hasn't come up in a long time. So what is a substrate? You see it here. The substrate is the substance that the enzyme reacts with. Let's highlight that. It's the substance that the enzyme reacts with. Okay, what's the product? See here? Here's the product. So how do we give the definition of a product? A product is what is formed when the enzyme reacts with the substrate. Okay, brilliant. Now, we're just going to go through some basic facts that come up a lot in your Leaving Cert about enzymes. These are all things that have been asked. Okay, now, I always ask this question to my students, right? I even did this chapter over Easter. I did this chapter over the Easter revision courses. So for some people here, I've done this about 4 times since January. But if you look at this here, right? I always say to my students, can anybody tell me who's here today? Name me a structural protein. Name me a protein, all right? So, name me a protein. Finn, sir, I'm late. What's an enzyme? Finn, come on now. You're late and you're coming into a class with hundreds of students and you're asking me what's an enzyme. Open up the first page. It's a biological catalyst. So have a look, all right? It's a biological catalyst. Now, everybody, name me a structural protein, all right? Name me a structural protein. So it's keratin. Brilliant. Well done. Oh my goodness, so people know that. But that's a nice one. But then I asked the students then this question. Name me a protein that's got a metabolic role. And people stop and say, I don't know. But this is from your food chapter. So can anybody name me a protein that's got well done on that answer, everybody? My goodness, so many people know that answer. But could anybody name me a protein that's got a metabolic role? Name me a protein that's got a metabolic role. And of course, the answer is an enzyme. An enzyme is a protein. It's not got a structure role. It has a metabolic role. Because enzymes, well done. Why? Because enzymes control metabolic reactions. You know this from your food notes. This is all in your food chapter. My goodness, the answers are flying in today. Excellent. So an enzyme is a metabolic protein. So if you're asked in your Leaving Cert in the food chapter, name me a protein that's got a metabolic role, enzymes is a good answer to go with. Why has it got a metabolic role? Because it controls metabolic reactions. What metabolic reactions, what chemical reactions do enzymes control? Enzymes control respiration. It's a chemical reaction. Enzymes control it. We'll study that tomorrow. Enzymes control the dark stage of photosynthesis. We'll study that on Wednesday. Enzymes control digestion. Now be very careful. In 2014, they asked you this. Name me a reaction that enzymes control in a cell. Which one of them, do enzymes control all three of them? But which one of those reactions doesn't occur in a cell? Digestion. Digestion does not occur in human cells. So be careful. If they ask you to name me a reaction that enzymes control in a plant cell or an animal cell, you go with respiration or photosynthesis. Don't go with digestion. Enzymes control digestion, but it's not in a cell. Okay? Now, if we then go down a little bit further, and then we're going to look at some exam questions then. So let's go down a little bit further. So if you then look at this here, you need to know what a catabolic reaction is. A catabolic reaction is what? It breaks down a larger substance into a smaller one. A larger, more complex substance into a smaller or simpler one. That's what a catabolic reaction is. Now, if I asked you, name me a catabolic reaction, you would say respiration or digestion. But I'm not asking you that today. I'm asking you to name me a catabolic enzyme. So a catabolic enzyme is an enzyme that's involved in breaking something down. I just said digestion is a catabolic reaction because it's breaking down food into smaller pieces. So therefore, any enzyme that's involved in digestion, therefore must be a catabolic enzyme. So, amylase, or you can see the ones from the previous page, lipase, protease, see them three enzymes there? Those three enzymes are all catabolic enzymes. Why? Because they're all involved in breaking down food into smaller pieces. So you could have went with any one of those. So just note there, amylase is a catabolic enzyme. But please, everybody, if they ask you to name a catabolic enzyme, you'll say amylase or lipase or protease. But if they ask you to name a catabolic reaction, then it's digestion or respiration. Don't get the two mixed up. Now, and you can see, look, amylase breaks down starch into a smaller substance called maltose. So that's why it's a catabolic enzyme. Okay? Now, let's look at the word anabolic. You need to know an anabolic reaction converts a smaller molecule into a larger molecule. That's what an anabolic reaction is. It converts a smaller molecule into a larger molecule. So, if you're, I see a lot of people writing in answers here. So here's another one for you. Could anybody name me an anabolic reaction in plants? And could anybody name me an anabolic reaction in animals? So could you name me an anabolic reaction in plants and an anabolic reaction in animals? There's one that does both, by the way. There's one common to both, but some people might know another one as well. So name me an anabolic reaction in plants. Name me an anabolic reaction in animals. So photosynthesis, well done, in plants. What about animals though? What about animals? What's the anabolic reaction in animals? Cells. Name me. I see a lot of people writing photosynthesis. No, respiration, Omar, is catabolic, so that would be incorrect. So, well done. I see the other one coming in. So, well done. So, everybody, an anabolic reaction in plants is photosynthesis or protein synthesis. An anabolic reaction in animals, protein synthesis. Protein synthesis covers both, but obviously photosynthesis is in plants. But both are, so let me repeat that. Photosynthesis and protein synthesis are both anabolic reactions. Respiration is a catabolic reaction. Now, the reason why, that's fine. I'm not, to be honest, that's got to do with a different chapter. The reason why I'm pointing that out is because if they ask you to name an anabolic enzyme, make sure you give an enzyme. So an enzyme that's an anabolic enzyme is DNA polymerase. Or some people this morning was there and we were studying RNA polymerase. They're anabolic enzymes or DNA ligase. These are all got to do with the DNA chapters. So note, that's a hard one. People don't know that one that well. So pick one of them and learn one of them as your anabolic enzyme. Remember, an anabolic reaction, protein synthesis or photosynthesis. But an anabolic enzyme, make sure you name the enzyme. No, digestion is obviously in your mouth or your stomach, so that's why it's not in a cell. So if they ask you to name a reaction that enzymes control in a cell, you got to go with photosynthesis. protein synthesis or respiration. Digestion is not in a cell, it's in your mouth or your stomach, okay? Brilliant. So look, what we're going to do, we're going to do a little bit more, and then we're going to spend a bit of time doing some exam questions, all right? So we're still on the beginning of this chapter, but we're doing so well here. We're flying through this. Okay, now, we said temperature and pH affect enzyme activity. Okay, let's look at temperature first. So if we look at temperature, human enzymes, they work best. Their optimum means what they work best at. Their optimum temperature is 37 degrees Celsius. You need to know that, okay? And a plant enzyme's optimum temperature is usually 20 to 30. You need to know that, okay? Now, the next thing then you need to know is this. You need to be able to draw the graph that shows the relationship between temperature and rate of enzyme activity. Now, when you're drawing this graph, please put the arrows in just like I have there to show it increasing, okay? And you label your both axes, of course. Now, look at the relationship. You need to be able to describe the relationship between temperature and how it affects enzyme activity. And let's look at this. So as the temperature increases from 0 upwards, what happens to the enzyme's activity? It increases, but then it decreases. There it is. So look here, everybody. As the temperature increases, the enzyme's activity increases, but then decreases. That temperature there is the optimum temperature. That's when it's working at its best. So that's all you need to do there. To be able to label both axes, to be able to draw the curve, and also to be able to write the relationship into a sentence. And you'll see the sentence here. As the temperature increases, the enzyme's activity increases, but then it decreases. People struggle with that. Something like that came up in 2014. Most people couldn't do it. And I'm going to show you that question. We're going to spend 5 minutes looking at some exam questions in a minute. But let's go through a little bit more here. Now, if you then look at this here, if the temperature is too high, an enzyme becomes denatured. Now, what does denatured mean? Denatured means an enzyme has lost its shape and it's lost its function. Now, in 2011, you were asked for 2 features of a denatured enzyme. They were basically just looking for the definition. Lost its shape and it can no longer function. Now, can I ask everybody, all right? Can I ask everybody, Lara, can you scroll up a tiny bit, please? Oh, I'm not too sure what you're referring to there, Lara. So, can I ask everybody there, can anybody name me, so now we know what a denatured enzyme is. Can anybody name me one factor that denatures an enzyme? Could anybody name me one factor that denatures an enzyme? So could anybody name me one factor that denatures an enzyme? Could anybody name me one factor? So could I say when it gets to optimum temperatures when it's working at its best? So when you refer to the word optimum, make sure you say it's working at its best or its maximum rate. All right. Okay. Thank you, everybody. Okay. Most people got 0 marks there. If I ask you, this came up last year. All right. This came up last year. If I ask you in your Leaving Cert this year, name me a factor that denatures an enzyme, and you say temperature or you say pH, you're likely to get 0 marks because temperature does not denature an enzyme. Because look at this. That's the optimum temperature. That doesn't denature an enzyme. So most people lose the marks here. So in last year's Leaving Cert, if you said temperature, you got 0 marks. You had to say extremely high temperatures or extreme heats. That's what denatures an enzyme. So let me repeat that. Now, hold on a second. If they ask you, name me a factor that affects enzyme activity, temperature, pH affects enzyme activity. But if they ask you at the question, name me a factor that denatures the enzyme, then you got to say something more precise. Extreme heats or extreme temperature. Hold on. Some people said extreme temperature, 0 marks. Extreme temperature could be 0 degrees. 0 degrees does not denature an enzyme. Okay? So last year was the first year that they were so harsh on that. So again, extreme temperature may not be good enough. Unsuitable temperature may not be good enough. Last year other years you might get away with it, but last year you had to say extreme heat. Because extreme heat means very high temperatures. Okay? If you go, or boiling, that would be okay. But if you went with extreme pH, that's fine. Or very high or low pH is fine. But in terms of temperature, you must say extreme heat. Or very high temperatures. Okay, I'm glad we clarified that because a lot of people got caught out of that last year. Okay, let's look at the second factor that affects enzyme activity. Sarah, unfavorable temperatures usually gets you the marks, but not last year. Extreme heat. Unfavorable temperatures might be 5 degrees Celsius, but that's not going to denature it. It's going to slow its activity down. So going by last year's marking scheme, they were very harsh on that. And I've never seen him as harsh now, so please be careful there. Okay, let's look at the second factor. The pH is another factor that affects enzyme activity, right? So, at low or high pH values, the enzymes lose their shape and become denatured. So again, you need to be able to describe the shape of the graph. pH on the horizontal axis, rate of enzyme activity, draw your arrows showing them increasing, and look at this here. As the pH increases, the enzyme's activity increases, but then decreases. Brilliant. Okay? Brilliant. Now, if you then look at this here, most enzymes, their optimum pH, they work best between 6 and 8 in terms of pH. But that's not all. So there is exceptions. Like, for example, there's an enzyme in your stomach that works best, called pepsin, that works best at pH 2 because of the hydrochloric acid in the stomach. Okay, brilliant. Now, we're ready. We're well through this chapter already, everybody, because it's so easy to study this chapter when you know what you have to study. So let's now go and open up our H1 question bank. Now, for students who are here today for free, all right, the H1 question bank is on the news feed there, all right? And I've for a lot of students or even premium members, I know a lot of you haven't got it, so because we ran out of copies. There was about 1,500 to 2,000 of the copies to hand out, so you'll see it on the news feed there as well. Okay, now Hannah, can't you say high temperature, or do you have to say extreme? Last year was extreme temperature, extreme heat, not extreme temperature, extreme heat. Extreme temperature is not the same as extreme heat. As I said, extreme temperature could be 0 degrees Celsius. That's extreme temperature, but that 0 degrees Celsius does not denature an enzyme. So you got to emphasize it's the high temperature. Very high temperatures or extreme heat. Same idea. Okay? Now, if you then look at this here, let's look at some questions. So this is on page now, on our question bank, page 50. Let me point this out. If people that are new here today, I'm saying this here to everybody, especially for those who aren't aware of this. If you know all these questions in this question bank booklet, then and you'll have sat a good chunk of your Leaving Cert before you even sit it. And what I mean by that is this. I won't say every question, but I will say the majority of the questions that are going to come up in your Leaving Cert this year, the majority of those questions are going to be in this question bank. Now, they might not be there word for word, but the idea of the question will be there. And that's why, like I told you, 57, 59, 61% of my students over the last 3 years got a H1 in biology. And I taught over 350 students, each one of those, in a certain day school, all right, in Dublin. But my point I'm making is they knew these. And when I asked them, what do you think of the paper? It looked really familiar. And I said, why do you think it looks so familiar? And they always tell me, because we've seen all these questions already from the question bank. Okay? Now, if you look at this, everybody, let's look at some exam questions. This is a really popular one. So you'll see here, all right? So I'm not going to do all the exam questions today because I've got pages of them here from previous papers. But here, look, you see here, what's an enzyme? It's a biological catalyst. What biomolecule is an enzyme made from? It's made from a protein. I love this one here because it ties in food with enzymes. Apart from carbon, hydrogen, and oxygen, there's one other element present in building block of enzymes. Well, we know enzymes are proteins. And what are the 4 elements in proteins from your food chapter? Carbon, hydrogen, oxygen, and nitrogen. So that's what makes them different, okay? Now, where in a cell is an enzyme made? Came up in 2021, ribosomes. What's the shape of an enzyme? It's folded. Some people say 3D. You never really see 3D on the marking scheme. I always like to say folded, okay? Now, we'll jump on down here. In 2023, they asked you to name the products out of the following reactions. So amylase breaks down starch onto maltose, protease breaks down proteins into amino acids, lipase breaks down lipids into fatty acids and glycerol. Now, All you had to do is name the products. But in that question, make sure you state which product is related to what enzyme. Okay, so just don't write them down, the 3 products randomly. Make sure you say which one belongs to which. And then you get your 9 marks, 2.5% of your paper. Okay, we'll go down here a little bit further. Okay, we'll go down here a little bit further. Look at this one here. Question 12. Name a protein that has a metabolic role. A lot of people got that earlier. An enzyme. An enzyme has a metabolic role. Okay, because enzymes are proteins and they control metabolic reactions. Okay, complex 3D shape, George. I think that would be okay. But folded is just to say, folded is just, proteins become folded, and we studied that in DNA and RNA this morning, and that allows them to become functional. Okay, now if you then look at this, Angela, for question 7, could you say for question 7? There's lots of options there for question 7. I didn't look at question 7 today. As I said, you'll do a lot of these in your own time because there's so many, but I'm going to cover as many as possible in the 2 hours. Look at this here. Question 13. Name 2 metabolic reactions that are controlled by enzymes. Well, enzymes control protein synthesis. Enzymes control photosynthesis. Enzymes control respiration. Those are 2 metabolic reactions, or there's 3 there. Now, in your leaving cert, if they ask for 2, I give 3 there. Never give 3 if they ask for 2. All right, I'm only revising today, so that's why I have all the answers there. But in your leaving cert, only give 2 when they ask for 2, because if you give 3 and they ask for 2, and one of the 3 is wrong, that will cancel out one of your right answers. So please only give the number that they require. Okay, so if you then look at this here, name one example of an anabolic enzyme and one example of a catabolic enzyme. RNA polymerase or DNA polymerase. All right, you can see they're opening both for there. So you can use either one of them as your answer because we study RNA polymerase in the DNA chapter this morning as well because it's involved in making mRNA. But there you go. A catabolic enzyme, you see the 3 options there. Okay, if we then go down to here, look at question 15. Name 2 processes that occur in plant or animal, look here now, cells that require enzymes. Well, enzymes control digestion, but that's not in a cell. So you have to go for protein synthesis, photosynthesis, or respiration. It said plant or animal cells, so you could go for any of those. Okay, now look at question 16. State 2 factors that affect the activity of an enzyme, pH or temperature. Okay, that's one way. But then look at this here. State 2 factors that denature an enzyme, extreme heat, or very high or low pH. So you see the difference between 16 and 17. Question 16 asks you 2 factors that affect enzyme activity, pH or temperature. Question 17, give 2 factors that denatures an enzyme. Then you have to say extreme heat or very high or very low pHs. Okay, we're going to keep on going down here. So I want us to then go down here a little bit further. So if you look at question 20 came up in 2013, or was it 2014? I think it was 2014 actually, but it might be 2013. We won't argue about it. It was around then, right? 12 years ago. So it says, all right, if you look at this question here, it says, study the graphs on enzymes and answer the question below. So here's the question A. Here's the question. In the case of each graph, state the relationship between the rate of the reaction on the y-axis and another factor on the x-axis. Now, I remember that year. It was 12 years ago. And I remember that summer's evening in June or afternoon in June when people came out of the biology exam. And I asked my students, what do you think of the enzyme question? And they said, hated it. Because it was never asked like this before. But it's so easy when you actually break the question down. So the first thing you do here is you label the axes. So they said the rate of reaction on the y-axis. So obviously this is the y-axis, the rate of reaction. Okay, now does anybody know what I'm going to label the x-axis is? So what am I going to label the x-axis? Tell me what I'm going to label the x-axis is, everybody. Ellen, DNA polymerase is an anabolic enzyme because it joins small pieces of DNA to form larger pieces of DNA. All right, so it helps to form larger pieces of DNA. That's why it's an anabolic enzyme. Yeah, DNA ligase as well. Fine, perfect, good. Okay, now, so people are saying temperature, pH. But the answer there would be no, because it tells you to label the x-axis as a factor. They didn't say which factor it was. Caitlin, well done. So you just have to call it a factor. They did not say which factor it was. So now we've labeled the axes, then this is quite straightforward. Let's look at the answer, everybody. So for graph A, as the factor increases, so look, as the factor increases, the rate of the reaction decreases. That's all you had to say. But look at graph B, it's the same labels. So look, factor here, rate of reaction here. Okay? Now, if you look at this one, as the factor increases, the rate of the reaction increases, but then it decreases, just like the one in your notes. So for a really difficult question that you first of all look at it, it's actually quite simple when you break it down. Okay, so I'll just give everybody a moment there. I'm just going to grab a sip of water. And then we're going to keep moving. All right, we're going to keep moving. So if you actually, I'm going to do one more because I would be annoyed if I didn't ask you this one and it came up. If you look at this question, this came up in 2014 as well. Question 21. Washing powders contain enzymes similar to the enzymes that are in your digestive system. Now, I would say then to my students, what do enzymes do in our digestive system? They break down food. So then the question is, why are such enzymes in washing powders? Why do we put enzymes in washing powders that are also found in our digestive system? Well, enzymes in our digestive system break down food. So these enzymes do what in washing powders? They break down food stains. A lot of people said they break down stains and got 0 marks because it's digestive enzymes that are in your digestive system. These enzymes do what? They break down food stains. And then it says, why is 40 degrees Celsius the recommended temperature for washing powders? Well, that must be the optimum temperature for these enzymes. And then it said, in 2014, what would happen to these enzymes at 80 degrees Celsius? Well, of course, that's an extreme heat, so therefore it would become denatured. Okay, everybody, let's go back. And there's a lot of other questions there that I didn't do because we've only got so much time today, but we're now going to go back and we're going to look at page 121. We're flying here, everybody. This may not even take 2 hours. We'll see how it goes. We might get finished even a bit earlier, but we won't rush it. We'll do it really well. Everybody, because what I want for you in these 5 classes is I want you to walk away saying, I understand this now so well. The only thing I need to do is really just study the definitions because I understand everything else. That's the way I want you to feel. I want you to feel that when you walk away from today, say, God, I understand that now. This is only going to take me like 20 minutes or an hour to study, whatever it is. Okay? Now, if you then look at this here, could you say the shape of an enzyme is globular? I have it in my notes as globular or folded. I like the word folded better. It's the one they use in the market scheme. Enzymes are globular proteins, which means they're folded proteins. Stick with the word folded. It's the more precise answer. Okay, now, if we then look at this here, everybody, we need to look at this now. We already touched on this briefly already, but let's just, earlier on in the chapter, but let's now look at this. So, if you look at this, everybody, the active site. The active site is, we saw it at the start, and we'll see it here. So, let's imagine this letter E is representing the shape of an enzyme. Obviously it's not, but enzymes are folded in shape. But let's imagine that did, right? So what part of the enzyme is the part of the enzyme that combines with the substrate, that changes the shape of the enzyme to suit the substrate? It's the active site. So put a star beside that, the active site. Now, we said a little bit earlier, enzymes are said to be specific. What does that mean? It reacts with only one particular substrate. Okay? All right, now, I see a question there. What is the best way we can learn and study definitions? To be honest, the one thing about my definition, I have them all in your notes, but I also have them at the end of each chapter. So it's nice to see them all together, so then you can just go through them a little bit faster. To be honest with you, there's not many tips about it. You just got to learn it. It's one of these things that's a big part of your Leaving Cert. 15 to 20% is either directly or indirectly related to definitions, okay, for biology. Now, if we then look at this, everybody, if they ask you to describe the active site theory, or if they ask you to describe the induced fit model, or if they ask you to describe how an enzyme works, they're asking you the same thing. So sometimes they ask you to describe how an enzyme works. Sometimes they ask you to describe the active site theory. They're asking you the exact same thing. So these are the 4 things you need to say to get full marks to describe the active site theory, how an enzyme works. Number 1 is the second point. The active site of the enzyme changes shape to suit the substrate. That's going to get you 3 marks. Okay? The active site of the enzyme changes shape to suit the substrate. The next thing that's going to get you 2 marks is the next point. The enzyme and the substrate react. And you see it there. There's the enzyme and the substrate reacting to form an enzyme-substrate complex. It's so important that you use the word complex there. If you don't, you're not going to get your second 3 marks. So an enzyme-substrate complex forms. Yeah, Katie, flashcards are good for definitions, absolutely. But at the end of the day, you got to sit down and learn them. No matter if it's flashcards or what, you just got to learn them. All right? Now, look at point number 3. Point number 3 is this, everybody. What happens to the substrate? The enzyme turns the substrate into a product. So the substrate is now converted into the product and released. That's step number 3. Step number 4. Now, at this point, you probably have full marks because it's usually 9 marks. And you probably have 9 marks, 2.5% of your paper right now. Just in case it's worth 12, or just in case you messed up on one of the previous points, you got to say this as well. The enzyme, because it's a catalyst, the enzyme and the active site of the enzyme returns to its original shape and can be reused because it's a catalyst. That means the enzyme can be reused after again. And that's another 3 marks. So that, if it's 12 marks, you'll get 12 marks. If it's 9, then you'll get 9 and you'll have 3 marks to spare. Okay? Now, that's been asked many times. Over the last, I would like to say 10 years, that's been asked. I forget, I counted it there not so long ago. It's been asked maybe 4 times, maybe even more. But that's been asked a lot in recent years. Okay? So I'll show you that now before we look at our first of the experiments. So I'll show you this question here. You'll see it here in your question bank booklet. So see it here. Now in your question bank booklet, all right, you'll see here question. I'll go down here a little bit further. Sorry now. Now, if you look at here, page 53. So if you look at page 53 from your question bank, and if you haven't got it in front of you, it's on the platform. Don't worry. You can look at it after. But if you look at this here, and by the way, for those who are premium members, the solutions to all these are on the platform as well. Remember I said that. So for premium members, my advice is, when you're studying each chapter at this point of the year, have your question bank open, but have the question bank solutions open as well. Okay, so describe the active site theory. Describe how an enzyme works. The active site of the enzyme changes shape to suit the substrate. An enzyme substrate complex forms. The substrate is converted into the product and released, but the enzyme and its active site returns to its original shape and can be reused. There it is. Now, I'm not going to you can write that out in your own time. It's straight from your notes. That's copied word for word from your notes. So I'm not going to, I could give you 5 minutes to copy that down, but I don't think you'll be that happy about that because we want to get through the chapter and we want to know it well. But practice that question yourself. It's been asked, as I say, 4, maybe 5 times out of the last 10 or 11 years. Okay? Now, if you then look at this question here, question 32, it says, a current theory of enzyme action is illustrated in the diagram below. It says, identify what is represented by A and B. So, can you tell me what A and B, you see it here. This is an enzyme, and an enzyme reacts with the substrate A. What is B? Well, that's when the enzyme and the substrate react together, so it forms an enzyme-substrate complex. Can anybody tell me what C is? What's C there, is everybody? What's C? What's C there? Sophie, definitions, they don't have to be word for word, but you do have to hit the key points. There's not much flexibility with definitions, I'm going to be honest with you. There's not much. All right, you got to try to get them word for word as close as possible. There's a little bit of flexibility of a word or two here or there. Okay, well done, everybody. So C is the products. It is the products. Okay, excellent. Now, it then says question B. Well done, everybody. Oh my goodness, it's so busy, the live chat today. It's amazing. Well done. Now, look at this. Which theory of enzyme action is illustrated by the diagram? This is the active site theory, because the enzyme is reacting with a substrate to make a product. Now, look at question C. This was all asked, it was a 2021 or something, around the last 5 or 6 years. What type of metabolic reaction? There's only 2 types of metabolic reactions. It's anabolic or catabolic. Which one is this? Well, you're breaking a larger molecule, A, into 2 smaller molecules, because you can see it's split in half. So it's a catabolic reaction. Justify your answer. You're breaking larger molecules into smaller molecules. There's your justification. It's the definition of a catabolic reaction. Okay, look, we keep on going here. We'll keep on going. So, if we then look at this, everybody, we're now going to go and look at our first experiment. Now, I say this, and I'm going to pause for a second, and I'm going to show you this here. look at this here, everybody. Right? So if you look, and for anybody that's new here today, you might let me know, new members, because the current members should definitely know this because I talk about this every class. But if anybody's a new here, a free member today, just for the week, did you know that question 8 was on these 8 experiments? Did you know question 9 was on those 7 experiments? And did you know question 10 was on those 7? Let me know if you knew that, because from what I hear, a lot of people didn't know this around the club. They don't. Every year. It's been like this for the last 4 or 5 years. Now, the reason why I'm showing this again, look at question 9. Question 9 is based on 7 experiments. And 4 of them are in the enzyme chapter. So in the next, let's say 20 minutes, half an hour, we're going to be doing the first 3 of these. And we're going to do the other one then later on. But here's the great news. A lot of people saying no, some people saying yes. Okay, but the point I'm saying, the reason why I point that out, everybody, is because if you know question 8 is those 8 experiments, then you'll study those 8 experiments, and that's 7.5% of your paper. Question 9 is those 7, so you'll study those 7, there's 7.5% of your paper. Same with question 10, and you have to do 2 of those in your leaving cert, obviously. Now, back to this. The great news is, everybody, listen to this now. The next 20 minutes, I am going to do 3 of these experiments. You might say, God almighty, how is Wesley going to do 3 experiments in 20 minutes? Because all 3 of them are pretty much the exact same. So if you know one of these experiments out of these first 3, now the 4th one is very different. We'll do the 4th one at the end of the chapter, okay? We'll do that in the second half of the class. But if you understand that these 3 experiments are pretty much the exact same, except obviously for the factor that changes, but same method basically, then you're going to be so much more confident with this. So we're going to do, as I said, these 3 experiments. All right, and then we'll do the 4th one in the second part of the class. And that means then we'll have done 4 out of 7 experiments, which is more than a 50% chance of one of these enzyme experiments coming up as question 9. Let's look at these experiments. Now, excuse me. So if we look at this, everybody, let's look at page 122. Now, I'm going to take the first one a little bit slow because not everybody is maybe not everyone studied these recently. So, and the other reason why I want to take the first one a little bit slow is because if you know the first experiment and we do it really well, then the other 2 that we'll do afterwards, you'll know it because we studied the first one so well. Okay, so we're on page 122 if you're using my notes. Amy, at this point of the year, do you think we should just learn question 8 and 9 in experiments and leave question 7 experiments out? Look, Amy, I would always say try to know all 22 experiments because you just don't know on the day which questions could be asked in a very difficult way. So I would try to be aware of all 3 groups of experiments. But I suppose what I'm saying is the night before you're leaving cert, you won't have time to study everything. So what I would say is then, or maybe the week before even, then you might focus in on the 2 questions you might kind of spend a higher chance of doing. Some people might do it the other way. Some people might say, I know 2 of those questions really well, or 2 groups of experiments really well, question 8 and 9, but I'm not really happy about question 10. I'm going to spend my time making sure I know that one well. So everybody's different, okay? Anyway, let's get back to this. You will see this then, everybody. So the first 3 experiments that we're about to do are pretty much the exact same, except for one factor that changes, all right? So if you then look at this here. So Sarah, when you're asking about, let's focus on enzymes right now, everybody, and then don't worry, I'm going to give you time for questions about can I leave this out? Or how much might this be worth? Let's leave those questions till maybe the end and I'll answer them all for you. But let's focus on what we're doing now and I'm telling you, you'll feel so much better. Okay, Sarah, come back to that question a little bit later. Okay? Now, and Ella, will you come back to that one later as well? Now, if you look at this, everybody. So, we're doing 3 experiments that are pretty much the exact same. Okay? So, we're using the enzyme catalase in all 3. That enzyme catalase breaks down the substrate hydrogen peroxide. So that's going to be the substrate in all 3 experiments. The product then is what? The enzyme catalase breaks down the substrate hydrogen peroxide into the product water and oxygen. So that's going to be used in all 3 experiments. So when I see students coming into their leaving cert, not knowing that enzyme, that substrate, and that product, then I'm saying to myself, they don't know the 3 enzyme experiments. Because the 3 enzyme experiments are based on that. That enzyme, catalase, that substrate, hydrogen peroxide, and that product, water and oxygen. There is other ones, but the majority use this version, okay? Now, where do we get the catalase from? The catalase we usually get from celery, so highlight celery, okay? Now, if we then go down here, so the first experiment we're going to look at is the effect of pH on catalase activity. Now, what I'm going to do is, before we read through this, I'm going to show you the diagram. And yes, you do need to know that diagram. So let's go and look at this here on page 123. Okay? Okay, John, you said my teacher didn't cover really any of the experiments for question 9. I really don't like just studying the theory of the experiments because I find it much easier when I actually carry them out. What should I do? Well, John, the good news for you is, John, will you not listen to me, John, the 7 experiments we're doing today, the 7 chapters that we're doing this week. Oh my goodness, people, maybe you're missing John at the start. Let me just show you. John, these 7 chapters we're doing this week, those 7 chapters have all the experiments in question 9. So if you're here for all the classes, John, you'll see me covering all of these experiments because we'll be doing the 7 chapters for question 9. And you'll feel better then, John, I promise you. And John, we're going to see them now. John, these are very popular experiments, so I would definitely know them. Now, and you got to remember, John, and everyone else, experiments can come up anywhere in the paper. Just remember that as well. Now, let's have a look at this, everybody. So, this here is the substrate. So, do you see here in the diagram? All right. So, this here is the substrate. And everybody, when we get these 3 experiments studied, there's only one thing left in this chapter. It's the immobilization of enzymes. And we'll do that in the last 40 minutes. So, we're going to spend 20 minutes on this, okay? So, let's have a look here, okay? So, if you look at this, so you will see here, all right? So, the hydrogen peroxide is the substrate. Thank you, John, for your question. I'm glad you asked because it's always good to ask. So, hydrogen peroxide is your substrate. Your chopped celery is your enzyme catalase. So, that's your enzyme. Okay? Now, so that's your enzyme. So, celery contains the enzyme catalase. All right? Now, hydrogen peroxide is the substrate. Okay, so the whole idea of this experiment is, look, when you put the enzyme catalase in with the substrate hydrogen peroxide, the enzyme catalase turns the hydrogen peroxide, the substrate, into a product called water and oxygen. Okay, and look, we have to investigate the effect of pH on catalase activity. So we got to measure what do we measure to find how active the enzyme is? We measure the product. That's how we measure the activity of an enzyme. The enzyme, let me show you that again. So the enzyme, let me show you that again. So the enzyme breaks down the substrate into a product. So how do we measure the enzyme's activity? You measure the amount of product. So let's look at this here. You got the enzyme, catalase. You got the substrate, hydrogen peroxide. So what you do then is you pour the substrate, hydrogen peroxide, into your enzyme, catalase, into your celery, and celery contains the enzyme catalase. What will happen in here is this reaction. This catalase will now turn hydrogen peroxide into water and oxygen. But here's the thing, everybody. We cannot see oxygen. How are you supposed to measure oxygen if you can't see it? Now, there is ways, but how are you supposed to measure oxygen if you can't see it? The easiest way to do it is this. We use washing up liquid. So when you add the hydrogen peroxide, your substrate, into your catalase, your enzyme, water and oxygen forms. Well, the oxygen gets trapped in the washing up liquid and that forms foam or bubbles. So that's how we measure the enzyme's activity. You measure the volume of foam in 2 minutes or per minute. Now, foam is not the product. Foam is not the product. The product is water and oxygen. But we can't measure oxygen as a product because we can't see it. Well, there is other ways to do it, but this is the way we're doing here. So if we can't see oxygen, how are we going to measure it? That's why we use washing up liquid. The washing up liquid, so when you pour the catalase and the hydrogen peroxide, the enzyme and the substrate together, water and oxygen forms, the oxygen gets trapped in the washing up liquid, that forms foam. That's the same principle for all 3 experiments. What's different in them then? Well, let's go back here. This experiment is asking you to measure the effect of pH on catalase activity. Now, we already know the effect of pH on catalase activity. We studied the graph earlier. As the pH increases, the enzyme's activity will increase and then it will decrease. We know that, but we got to prove it. So this is what you do. Okay, so back to page 122. If you're measuring the effect of pH on catalyst activity, what are we changing? We're changing the pH. How do we change it? By using different pH buffers. If you say you change the pH by using a buffer, you get 0 marks. A buffer keeps the pH constant. How do we change the pH? By using different pH buffers. Okay, so the first time you do this experiment, all right, measuring the effect of pH on catalase activity. The first time you do it, you might use pH buffer 4. The second time you do it, you might use pH buffer 6. The third time you do it, you might use pH buffer 10. Whatever it is, right? The point I'm asking is, if they ask you what factor you changed the pH. If they ask you how you changed that pH, you use different pH buffers. So every time you do it, use a different pH buffer. Okay? Now, if you're changing the pH, then temperature must be kept constant. How do I keep the temperature constant? By using a water bath or the same water bath. Okay? All right, brilliant. Now, if we then go down a little bit further, okay? So if you then go down a little bit further, you will then see, all right? So look at it. And by the way, why should we only change one factor? Why is it important that you only change pH and not temperature? Because you're only allowed one variable. You're only allowed one factor to change in an experiment. Because if you change both, then you don't know which one you're measuring in terms of the product that's being formed, in terms of its impact on the enzyme activity. Now, let's now read through the experiment. Now, let me just throw this out at you, everybody. The last 2 years, question 8, 9, and 10, the 3 experiment questions, you had to describe the entire method, basically, right? Now, before that, that was unusual. But for the last 2 years, question 8, 9, and 10, you had to describe the procedure. Now, if you're asked to describe the procedure, I would include these first 2 points. We're changing the pH by using different pH buffers. We're keeping the temperature constant by using the same water bath. And then we dive into the experiment. We say, we added washing up liquid because the washing up liquid was trapping the oxygen to form foam. So that's why we used the washing up liquid. Okay? We then added celery because celery contained the enzyme catalase. Okay? So that's why we use celery, because the celery contains the enzyme catalase. We then added hydrogen peroxide using a syringe. Why did we use hydrogen peroxide? Because it's our substrate. It's our substrate. Perfect. Now, we made sure then everything was in a water bath at 25 degrees Celsius for a few minutes. So everything was in a water bath at 25 degrees Celsius because we're keeping the temperature constant. Okay? You're keeping the temperature constant. John, I'll come back to that question in a second. It's a great question. But, so what next? What we do then is, look, we pour the hydrogen peroxide into, which is the substrate, into your catalase, your enzyme. Okay? And so that's what you do there. You pour hydrogen peroxide into your catalase. What do you do then? This is the most important point. We now record the volume of foam in 2 minutes or per minute. Doesn't matter. Just say 2 minutes or per minute. Because a lot of people say you measure the volume of foam and leave it at that. You're not, if that's 6 marks, you're getting 3 out of 6. Because, and sometimes it is 6 marks. You must say per minute or in 2 minutes. And if you didn't mention it at the start, mention it now. You repeated this and you changed the pH by using different pH buffers. Not buffer, by using different pH buffers. That's the first experiment, everybody. That's it. Okay? So, and the results are, as you increase the pH, the enzyme's activity increases and then decreases. And that graph is asked so often. Now, what is the control in this experiment? The control is, all right, do not add chopped celery. And if there's no celery, there's no catalase, because that's where the catalase comes from. So if there's no enzyme, there's going to be no product. So therefore, there's no oxygen. And if there's no oxygen, you're not going to see any foam, because the oxygen gets trapped in the washing up liquid. They generally don't ask about the control on that one a whole lot, but just be aware of it. Now, that's the first one. Okay, let's now go and look at the second version. Now, as you go to the second version, John said a question there. Do you think because the new course is so focused on carrying out investigations? John, I honestly think they're trying to show what's going to happen in the new course, because the new course is very different to this course. And the 5th years now currently, they're going to have a lot more of that. So maybe they're trying to get us aware of that. So yeah, I would say, John, that could be one of the reasons why they've done that over the last 2 years. So do not be surprised if question 8, 9, and 10 are asking you to describe the procedures from start to finish again. Okay? Now, and if you are that, if that is the case, just bullet point form, simple key points, like my notes here. Now, let's look at the second one. And remember, everybody, look at this. Look at this. There's 4 experiments. There's 4 experiments from this one chapter. Okay? There's 4 experiments from this one chapter on enzymes, and we're doing 3 of them right now because 3 of them are pretty much the exact same. So if we then look at this here, so let's look at the second one. The great news is, everybody, very little changes. So let's have a look at the second one. The second one now is to measure the effect of temperature on catalase activity. Same enzyme, same substrate, same product. So that's all the same. So let's go down to the diagram. So your chopped celery contains the enzyme catalase. Your hydrogen peroxide contains is your substrate. So that's all the same so far. You're going to pour the hydrogen peroxide, which is your substrate, into the celery, which contains the enzyme catalase. The catalase will turn the hydrogen peroxide into water and oxygen. We got to measure the enzyme's activity. You got to measure the product. We can't see oxygen, so therefore the washing up liquid will trap the oxygen to form foam. So everything's the same, except what? You're changing temperature. Look at the title. You're changing temperature. So look this time. If the factor you're changing is temperature, but how? By using different water baths. Do you remember in the previous experiment? We changed pH, but look at this. In the previous experiment, we changed pH by using different pH buffers. And we kept the temperature constant by using the same water bath. It's the other way around this time. We're changing the temperature by using water baths at different temperatures, or different water baths. But we kept the pH constant by using the same pH buffer. Very important you get that wording right. Okay? But if you then go down here, so look, let's go through it. It's the exact same procedure, except we're changing different factors. Okay? So if you look at this one here, all right, we're using pH buffer. You don't even have to specify the pH buffer that you're going to use. But if you want, you can. Using pH buffer 9, and that's going to keep the pH constant at 9 because we're changing the temperature. The pH has to be kept constant. So we're using the same pH buffer. Now, the celery, we're adding celery. Why? Because the celery contains the enzyme catalase. We're adding washing up liquid because it traps the oxygen to form foam. Nothing's different so far except you're changing temperature by using different water baths at different temperatures, and you're keeping the pH constant by using the same pH buffer. Okay, that's the only thing that's different. Using a syringe, we're adding hydrogen peroxide to our beaker, that's our substrate, and we're using this time, look, water baths at different temperatures. That's how you change the temperature. So the first time you might have an ice cold water bath, maybe 0 degrees Celsius. The next one you do 10 degrees Celsius. The next one 30. The next one's 50. Whatever it is. They don't care. They just want you to know how you change the temperature. You change the temperature by using water baths at different temperatures. Now, when the time is right then, you pour the hydrogen peroxide into the catalase, into the enzyme. Really important then, same as the last time. You record the volume of foam in 2 minutes. You recorded the volume of foam in 2 minutes. That's how you measure the enzyme's activity, or per minute. Just mention some sort of time interval there. Okay? That's the experiment. You repeated the experiment different times by using different temperatures in different water baths. Okay? Excellent. And the results are this. We saw this graph before. As you increase the temperature, okay, the enzyme's activity increases and then decreases. The exact same graph we saw earlier when we talked about that graph. Just make sure you know how to label the axes. Don't lose silly marks. Okay, do not lose silly marks there. And there it is. That's the second experiment. Now, before we do the third one, that's pretty much the same. We'll do a couple of exam questions first of all. So I want you now to turn to page 54 in your question bank. And I just see a couple of questions there. Lisa said, Wesley, you refers to, okay, Lisa, if this is obvious, but I can't find segregation cross. So Lisa, the genetic cross videos, Lisa, There's 2 chapters I still have to put on the platform fully: genetic crosses and plant reproduction. But Lisa, those videos I did them live. They're up in the Sunday masterclasses. Go to Sunday masterclasses and you'll see it there. There's 4 hours on genetic crosses there. Okay. Now, but all the other ones there on demand. Now, look at this. Thanks for asking. Now, look at this, everybody. So we did 2 experiments so far that are pretty much the exact same. Okay, so look, we'll do a couple of questions here. The effect of pH on catalase activity. These are the questions they've asked on this before. Name the enzyme, catalase. Name the substrate, hydrogen peroxide. Name the product. The product's not foam. The product is water and oxygen. Where do we get this enzyme from? What's its source? Celery or liver. Most people use celery though. Okay, I won't run through them all, but look at this one here. If you're changing the pH, look, what factor do you vary? pH. How do you carry that? By using different pH buffers. Question 39. What factor do you keep constant? Well, if you're changing the pH, you're keeping the temperature constant. How? By using a water bath. Okay? All right. Now, if you then look at this here, Tom, or who's out there? Angela. Yeah, you can draw that a little bit narrow, the pH one narrow as well. But it's the same idea. It just depends how you plot it along the horizontal axis. So either way, it's the same idea. So Angela, don't worry. So if you then look at this one here, question 42. How do you measure the enzyme's activity? You measure the volume of foam, but that might not get you full marks. In 2 minutes or 1 minute. Just mention a time interval there. Okay? Now, if you then look at the graph, there's your graph. The rate of enzyme activity, pH, so you show it increasing like so. All right? Perfect. Nothing difficult there. Now, be aware. Know how to draw the diagram of the equipment. I see so many people making that mistake, by the way. If they ask you to draw the diagram of the equipment, don't draw the graph. You only draw the graph when they ask you to draw a graph. You only draw the equipment when they ask you to draw the equipment. So please make sure that you see that, okay? So if we then look at this here, if we then go down here a little bit further, question 48. So question 48 here. If you're asked to describe how you carried out the investigation, and as I said, this is the type of questions come up in the last 2 years. This experiment has not come up, but my point is this is the type of question that has been asked. Describe the procedure. So there's the procedure described for you. And I'm not going to run through it again because it's straight from your notes. Don't panic if you don't write everything in, but what I would always include is this. Make sure you name the substrate and the product. Make sure you name the enzyme. Make sure you explain how you change the factor, pH, by using different pH buffers. Make sure you state how you keep the temperature constant by using the same water bath. Make sure you state you poured hydrogen peroxide into your catalase. Make sure you state, okay, how you measured the enzyme's activity, measuring the volume of foam in 2 minutes. And what I do notice is this, everybody. Always include a safety procedure when they're asking you to describe the procedure. They'll often award marks for a safety. So always include a safety procedure, no matter how simple it might be. And look, I don't actually have it there, but always throw in the control. I should have a control there as well. But always, so if you're asked to describe the procedure, describe the procedure, but also include in that procedure a safety precaution and your control. And then you'll be absolutely safe. Now look at the second experiment we did today. The majority of the questions are the exact same. The only one that's different, the only one that is different is this one. Look at this one. This is the only question that's different compared to the other one we just done in terms of pH. This is temperature this time. The only thing that's different is this. What factor did you keep constant this time? Well, if you're changing temperature, you keep the pH constant. How did you keep it constant? By using the same pH buffer. Okay, and then obviously what factor did you vary? Well, you're varying temperature. State how you varied it. And I forgot to state how. How do you vary the temperature? Water baths at different temperatures. Okay, perfect. Now look at this. Why is it important that only temperature was varied? Well, you only should have one factor. Only one factor should be changing. You should only have one variable. Only one variable there, everybody. Okay, so look. It's the same. You might say, look at this question. Same for temperature. Describe how you carried out the investigation. This procedure is pretty much the exact same as the pH procedure. Because it's pretty much the exact same experiment. The only thing that's different is this. If you're changing the temperature, so you're changing the temperature by using a water bath at different temperatures. So look, that's what you're changing, and you're keeping the pH constant by using the same pH buffer. The rest of that procedure is the exact same. Okay, now obviously the graph is a little bit different because you're having temperature there instead of pH this time. Okay, everybody, let's now go and look at our third one. Look, everybody, we're flying here. We're going to look at the third one now. The third experiment. Remember? Oh my goodness, I better go back and show you this because people forget this. Now look at this here. The third one is pretty much the exact same as the other two. Now it is a little bit more, it is slightly different. It's very similar, but it's the same idea, So let's go and have a look at it, everybody. Let's go and look at the third one. And fingers crossed one of these now will come up in your leaving cert and it'll be 7.5% of your paper. Okay, so let's look at the third one, everybody. Here we go. To investigate the effect of heat denaturation on enzyme activity. So this question is asking about denaturing an enzyme. Now we know what a denatured enzyme is. A denatured enzyme is an enzyme that has lost its shape and it can't function. So that's what they're asking you. If you denature an enzyme, how does it affect its activity? Well, it can't function. So there should be no product. So look, same enzyme, same substrate, same product. But obviously, when you're measuring the enzyme's activity, you're going to have zero foam this time because this enzyme, catalase, was denatured. So you're not going to get any water oxygen. If there's no oxygen being formed, there's no oxygen getting trapped in the washing up liquid. So you're going to see no foam. Now, I see a great question here. A great question about that. Hold on now, where was it? Is it important, all right, that if you shoot, like in terms of order, when you're asked, if you're asked to describe the procedure, is the order important? Try to get it in the right order, but they're not going to penalize you too much. Like it doesn't have to exactly bullet point by bullet point. Try to get it somewhat in the same order for your own sanity, for easier to put down, and it's easier for the examiner to correct as well. Okay, so the straight answer to that is, unless it's completely upside down, you'll be okay. So even if it's slightly out of order, you'll be all right there, okay? Now, but try to get it in the right order. Okay, so if you look at this then, so in this experiment, let me get my blue pen here, they're asking you to denature it. So how do we denature it? We boil it, everybody. So highlight boiling, all right? Highlight boiling, all right? Now, Lara, you said how many bullet points? It depends on your how precise you are in your write-up. So just, that depends on yourself, and you don't have that much room to write it, so try to be as concise as possible. Okay, so we boil the enzyme, everybody. Now look at this. If you boil catalase, then the enzyme's denatured. If the enzyme's denatured, you're going to get no activity. There's going to be no product. No product means no foam. So let's go down here. Look here. So look here, everybody. So I'll go right here so you see it better. So look, hydrogen peroxide is your substrate, just like the last two experiments. Your celery contains your catalase again. But look at this time, everybody. It's boiled. So it's boiled catalase. It's boiled. Let me write that a little bit neater so you can understand my writing. So one more time. So it's boiled catalase. So it's still catalase, but it's boiled. Now, let me pause here for a second, everybody. Let me go back here. What factors are we keeping constant in this experiment? Now, I should have said this at the start. A lot of people get heat denaturation mixed up with the temperature experiment. Temperature, you just change the temperature by using water baths at different temperatures. And you're seeing which temperature does the enzyme work at its best. It's optimum. But here, you're denaturing it. So it's not the same. You're boiling it. Okay? So the keyword there is denaturation. Now, what factors are we keeping constant? We're keeping the temperature constant by using the same water bath. We're going to keep the pH constant by using the same pH buffer. So in the first experiment, we changed the pH by using different pH buffers. And we kept the temperature constant by using the same water bath. And the second experiment, we changed the temperature by using different water baths at different temperatures. But we kept the pH constant using the same pH buffer. But in this one, we're keeping both constant. Now, I know people are saying at home, hold on a second. I thought we're boiling it. But this is the thing you got to realize. You're boiling the catalase, yes, to denature it. But then, once it's in the water bath, the temperature is constant from that point onwards. Because you're investigating by denaturing an enzyme, how does that affect its activity? So, you're boiling it to denature it. That's done before you even start with this diagram. So it's boiled before you put it in here. And when it's in here then, it's already denatured. And once it's in here, the water bath is going to keep the temperature constant. The pH buffer is going to keep the pH constant. So those factors, neither are changing this time. They're both staying constant. Okay? So that's what's make this one a little bit different. So this one's a little bit different to the first two. They're the same enzyme, same substrate, and same product in all three experiments. But in this one, you're boiling the enzyme to denature it. That's different. And you're not changing the pH and you're not changing the temperature. They're both kept constant. So that's what makes this one slightly different to the other two. So let's go through this method. We're using a pH buffer to keep the pH constant. You can mention a number if you want. Usually not expected, but you can mention it. You're going to boil the enzyme for 10 minutes to denature the enzyme. Okay? Now, this is really important one, this one, everybody. Put a star beside point number 4. At boil catalyst, that's your test. And on boil catalyst, that's your control into two separated graduated cylinders. I think it was 2019 they asked that. How did you treat the enzyme in the test? How did you treat the enzyme in the control? So many people couldn't answer that because they didn't even understand what the word test meant. That's the one you're investigating. So how did you treat the enzyme in the test? You boiled it. How did you treat the enzyme in the control? It wasn't boiled. Okay? Now, what do we do then? Same as the other. We put the hydrogen peroxide, your substrate, using a syringe into the beaker. Okay? We're going to keep the temperature constant using a water bath. Okay? We're going to pour then, look at this. Let me rub this out here to tidy this up a little bit. So, you're then going to pour the hydrogen peroxide, your substrate, into your catalase. Brilliant. All right. Then we're using washing up liquid as well to trap the oxygen to form foam. I said froth this time. Foam, froth, bubbles. Same idea. Okay. Then this is the same as well. You're going to record the volume of foam in 2 minutes to measure the enzyme's activity. So the foam, well, hold on. Does foam form in here? It depends. If it's the test, there'll be no foam because the enzyme was boiled, because the enzyme's denatured, so no activity. Because if the enzyme is denatured, there's going to be no activity, so there's going to be no products. But in the control, you're going to get foam because that enzyme wasn't denatured. So there's the result of the experiment. The boiled catalyst produced no foam or froth, whichever way you want to say it, or bubbles, because it was denatured, because it was boiled. The unboiled catalyst, it wasn't boiled, so it produced foam. So it's pretty much the same as the other two experiments, except this time you boiled the enzyme to denatured, and the temperature and the pH are kept constant. So that's slightly different that way, but it's the same idea of the setup. So let's look at some questions on this, and after this then we're going to have a 5-minute break, and then we're going to come back for the last half hour, and we're going to look at the immobilization part. Okay, and for some people it's the trickiest, so we'll get that, don't worry, we'll get a full half hour on that, we'll get that sorted completely. So let's have a look at this, everybody. So let's look at a couple of questions on the heat denaturation one. What is a denatured enzyme? It's an enzyme has lost its shape, can no longer carry out its function. So look, name the enzyme, name the substrate, name the product. That's the same in all three experiments. Okay? What makes this one different is, how did you denature it? Because we didn't do that in the other two. You boiled it for 10 minutes. Name two factors you kept constant. In the other two experiments, we changed the factor, pH or temperature. In this one, we're keeping both constant. How? pH kept constant by using the same pH buffer, not buffers, buffer, singular. And I kept the temperature constant by using a water bath. Now, look at this one. Describe how the enzyme was treated in the test and in the control. In the test, you boiled it. In the control, you didn't boil it. Okay? This was also asked that same year. Compare the results of the investigation in the test and in the control. The test, there was no foam produced because the enzyme was denatured. Because there was no product, obviously, because the enzyme was denatured. But the control, foam was produced because the enzyme wasn't denatured. Now, if they ask you to describe that procedure, don't panic. Most of it's the same as the other two. The only thing that's different this time is you boiled it for 10 minutes to denature it, And the other thing is different is, of course, is you kept the temperature constant. Remember, you boiled it before you put it into the water bath. But once it's in the water bath, the temperature is kept constant by using the same water bath, and the pH is kept constant by using the same pH buffer. So that's the only difference here. And of course, the results are different. And always include the results, everybody. Always include the results, unless they specify somewhere else in the question. So if you're asked to describe the procedure, we could always include a safety precaution, always include the results, and always include the control. But sometimes they ask that as a separate question, and obviously then you wouldn't. But if they don't, I would always include a safety procedure, your control, and the results, as well as the write-up, if you have the room to do it. Okay, everybody. Now, the only thing, there we go. The only thing now that we have to do is about 3 pages of information on immobilized enzymes. So I know I need a wee bit of a break here because I'm clamming up here. It's getting a bit warm. I must open a window. So what I'm going to do is don't log off. All right. All I'm saying is here, if you found that good so far, don't say to yourself, oh, I've done enough for one day. You're leaving certain 6 or 7 weeks time or whatever it is. Stay down for this next half hour. Let's get the immobilization part done. And then you can say, I've got this chapter down to a T. And I'm going to do the same now with all the other chapters over the next couple of days. So it's 7:25. Have a break for 5 minutes. Don't log off. Just stay where you are. I'm going to stay here for 5 minutes. If anybody's got any questions, you can ask away for 5 minutes. But in the next half hour, so we're going to be going again at 30 minutes past. In the next half hour, we're going to go and look at the immobilization. I'll spend 10 minutes on the theory behind immobilization, and I'll spend 20 minutes then on the experiment. And then that's it. So I see a couple of questions here. I'm just going to go back and answer a couple of questions. Go have a toilet break. Go have a glass of water, wherever it is. Jack says, if they ask you what product, the product is water and oxygen. Okay? If they ask you what the product, if the experiment is, should say what? So the product's the same. I know the enzyme's denatured in the test, but in the control, it wasn't denatured. So it's, the same product. So James says there, what would be the safety for these experiments? Goggles, like the usual. Like just, keep it simple. All right. James, can you make different pH buffers that use and do not have to start with pH buffer? James, they don't usually ask you what pH buffers you use. All they want to know usually is, how did you change the pH by using different pH buffers? That's it. They don't ask you which ones you used. Rachel, and as I said, for those, we're just going to give everybody a break here. So if you're not, go have a little break. Don't be worrying if you are not here for the questions. I'm going to be going at 1:30 again. Rachel, when asked for a safety precaution in any experiment, could you just name it, or do you have to say why as well? No, you normally just explain what the safety precaution was. I wear goggles, or I wore a lab coat to protect my skin, or whatever it is. Okay. What experiment would most likely be asked at a higher level? I'm not going to go there because there's so many, over the last 3 or 4 years, so many, the deferred paper, the Liebenstedt paper, so many variations of experiments have been asked. Just learn 8 experiments in question 8, learn the 7 experiments in question 9, have them down to a T, or learn the 7 experiments in question 10. Know 2 of those experiment groups really well. So no matter what is asked, you're not going to be caught out. Okay? Ash, Openit. I got a H4 in my biology mock. Because I didn't study for biology. I will say this to everybody. Do you remember I told you 61% of my students got a H1 last year? And that was 350 students I taught. And the year before was 59%. And the year before was 57%, right? Point I'm making is this. I would say a good half of them got a H3, H4, H5 in the mocks. And some of them I would have thought, do you know what? I don't think that student's going to get a H1. I would love for them, but I said, I don't know if they're going to get it. And they do. And the reason why is they know the questions from the question bank inside out. And they work hard on sections. So don't spread yourself too thin. Make sure you know your 7 chapters today. And I'll talk about it at the end, but you can have certain sections studied really well that will be worth a certain percentage, and therefore you'll get that top grade. I'll talk a little bit more about that at the end. Wesley, can you put the plant chapters on while you teach the human chapters during the biology blitz? Can you put the plant chapters on while you teach the human chapters during the biology blitz? What does that mean, Ash? Can I put the plant chapters on? What do you mean by on? Don't know what you mean by on there. Okay, come back to me on that, Ash, what you mean by on. All right, we have 2 more minutes to go, everybody, before we go again. Emer, do I have enough time until the Leaving Cert to cover all the systems as well as revise most of the other topics? You're talking about the human biology systems. Emer, if you're struggling with the systems, I don't know, Emer, if you're on the platform. Is that the Emer that's on the platform? Emer, you have 2 things. If you're struggling with the systems, you can use my on-demand because all of human biology is up there. Or you can attend my Blitz in 2 weeks' time, May the 10th, on the 5 big human biology chapters. And that'll get you go. But like, Emer, yes, you do have time as long as you get working hard every night now. And if you attend these 5 classes, that'll be these 7 chapters revised for you, okay? Ruby, what experiments do you think are you? Ruby, I said there, Ruby, I'm not going to go there with what just no question 8. No question 9 or no question 10. No 2 of those groups of experiments from start to finish. Because you don't want to get caught out. Just as simple as that. Alright? And if you're after H1, H2 or even H3, you want to make sure you have that. Okay? Let's just play. I still don't know what you mean, Ash. I can put the notes on the news feed. Is that what you mean? I mean, the class you caught on plants earlier in the year, can you replay them? Yeah, Ash, they're all on the platform. All the videos are on the platform. Some of the plants are on the on-demand, and then plant reproduction is on the platform is on the all the grinds. Every single grind is recorded, include these ones, of course. Okay, we've got 1 more minute before we kick off here, everybody. Clodagh says, I really struggle with question 10. If I knew question 8 and 9 inside out, will I be okay? Well, you should be, unless the experiments are asked in a hard way. But I would say to you, Clodagh, there's 3 or 4 experiments in question 10 that are nice. And use my videos on the platform if you're on the platform. I think you are, Clodagh, on the platform. Like the hard experiments. There's 2 hard experiments. Go and watch those experiments on the on-demand platform. And that's those 2. The leaf yeast experiment came up last year. So it might come up again. Have a look at that one. But then the plant structure, the dicot stem, my God, there's no simpler experiment on it. So you could still have 3 or 4 of those studied just in case, you know, and then maybe leave out some of the ones that you're not happy with on question 10. Okay, Anna, one last question. Are you able to put up some of the on-demand videos on human defense system? Anna, the general defense system video is up there. I know you're looking for the second half of that chapter, the specific defense system. I will, Anna. That's one little thing I have to finish off there as well. But Anna, I taught that over 75 minutes about 4 weeks ago. And it's on the live grinds as well. So just go into, I think it's under Jennifer's because it was a Tuesday one. And you'll see it there. Okay? You'll see it there. But yes, I'll get that up there as well. I'm flat out at the minute with classes, but I will, I promise you. All right, let's get going, everybody. Let's go on to immobilization. So we're now going to page 127. Any other questions? Hold off to the end in terms of layout of the paper, and I'll answer them at the end. Admin, you might give me a few extra minutes at the end, so we're not, because I think I'll have a lot of people here today, so I want to make sure I answer everybody's questions. And if I've missed any of your questions during the class, you can ask them again at the end. So admin, you might stick it on an extra 15 minutes at the end, just in case they need time to answer more questions. Okay, everybody. So admin is just the person who's in control of the live chat there. Okay, so let's look at this, everybody. Immobilized enzymes. So let's look at immobilized enzymes. Immobilized enzymes, what do you mean by it? An immobilized enzyme is an enzyme that's attached to each other. Or it's an enzyme that's attached to an inert material. Maybe a gel, like sodium alginate. Okay? So let me just show you this here. Let's go over the next page. These enzymes here, that I'm going to circle, they're in blue or red, whatever you want to call them. Those enzymes are attached to each other. Those enzymes are therefore immobilized. Here's another way to do it. These enzymes here, that I'm coloring in blue, or red, that were blue, those enzymes are trapped in a gel called sodium alginate. You need to know that gel. Okay? Because those enzymes are trapped in a gel, those enzymes are immobilized. Now, why do you want to do this to an enzyme? Why would you want to trap enzymes to each other, attach enzymes to each other? Why would you want to trap an enzyme in a gel? Why would you want to immobilize an enzyme? Here it is. It's very simple. Because if we use enzymes in industry, we use enzymes to break down substrates to make products, like food products, medicines, and so on. There's a long list. So we use enzymes to make things for ourselves. But if we're going to use an enzyme in an industry, if we're going to put an enzyme into this bioreactor, we should immobilize it before we put it in there. Now, why? Well, let me just catch up on this a wee minute first of all. So we use enzymes in bioprocessing. What is bioprocessing? It's the use of an enzyme in chemical reactions to make a product. So we know an enzyme breaks down a substrate into a product. So, and we know enzymes do that in our body. Enzymes carry out allow respiration, photosynthesis, digestion, protein synthesis. We know enzymes are involved in all of those processes. And that's fine. But what now we're talking about is different. We also use enzymes in industry to make things for ourselves. And we refer to that as bioprocessing. Now, what products are made using enzymes? We make antibiotics using enzymes. We make vitamins using enzymes. Now, here's the thing. If we're going to use an enzyme, if we want to use an enzyme to break down a substrate to make a product in industry, we've got to be able to control the environment that the enzyme has been put into. So we use a bioreactor. A bioreactor is a container. What's in that? What's happening inside that container? Chemical reactions. Who's carrying them out? Enzymes. So we're putting enzymes in here. All right. The enzyme carries out a chemical reaction in there to make a product. The enzyme will break down a substrate that we add in there as well to make a product. Now, why do we use bioreactors to do this? Because we can control the pH, we control the temperature. Bioreactors those containers allow you to control those things so enzymes can work at their optimum rate. So the enzyme we're going to put in here today is sucrase. That's the enzyme. The substrate is sucrose. Okay? Now, how are you not going to... This is the one we're going to use... This is the one we're going to use in the experiment as well. So we might as well use it here as well. So the enzyme is sucrase. Remember, the enzyme always ends in the letters ASE. Okay? The substrate is sucrose. All right, everybody. So we're going to put that enzyme and that substrate into that bioreactor. Now, at the end of the reaction, the sucrase will break down the sucrose into a product. So the product in the bioreactor is glucose and fructose. I'll just write glucose for today, but glucose and fructose. You'll see it in my notes further down. Okay? Now, here's the thing. I'm going to ask you all here now this, all right? And hopefully now we'll be okay with this. If I put this enzyme sucrase and the substrate sucrose into that bioreactor, the sucrase, the enzyme, will break down the sucrose, the substrate, into the product called glucose and fructose. My question to you is this. After the reaction has taken place, when you put sucrase, the enzyme, and the substrate sucrose into the bioreactor, the enzyme sucrase turns the substrate sucrose into the product called glucose and fructose. At the end of the reaction, what's inside the bioreactor? Well, obviously the product is in here. Is there anything else in the bioreactor at the end? So we're putting the enzyme and the substrate into the bioreactor. The enzyme turns the substrate into the product. So the product now has been formed inside the bioreactor by the enzyme reacting with the substrate. So the product's in the bioreactor at the end. What else is in the bioreactor at the end? All right, what else? I'll give you a second. Well done. The enzyme is also in there. The enzyme is also in here. Now, we do not want the enzyme and the product to be in there. Not sucrose, everybody. Sucrase, just the enzyme, right? So, we do not want the enzyme and the product. Let me tidy this up a little bit now. Okay, so let me just tidy that up so it doesn't look too messy. So, look here. So, what's in there? One more time. The enzyme and the substrate are both put in here, all right? But at the end of the reaction, the enzyme and the product are what's now in the bioreactor, because the enzyme is a catalyst. It speeds up the reaction, but it doesn't get used up. Now, here's the thing. We do not want the enzyme and the product in the bioreactor at the end together. We want to be able to take the enzyme out. And by removing the enzyme, then you're left with the product. Okay? But the only way to take the enzyme out of the bioreactor is if what? If you've immobilized the enzyme. Because look, everybody, if these enzymes are all attached to each other, just like that, if those enzymes are attached to each other like that, okay, that means if you take out one, you're taking out all the enzymes because they're all attached to each other. Or if the enzymes are trapped in the gel, just like that, by taking out the gel, you're taking out the enzyme. So that's why we use immobilized enzymes. Because if you put an immobilized enzyme, let's say that's trapped in the gel, into that bioreactor. So imagine you put sucrase, but the sucrase was trapped in the gel. That enzyme was trapped in the gel. Then you can take it out afterwards. And the product then is all by itself. So that's why we immobilize an enzyme, everybody. So now I want you to highlight this heading. What's the advantage of immobilizing an enzyme? The advantage of trapping an enzyme in a gel is the enzyme can be removed from the bioreactor. That's the first thing. The enzyme can be removed from the bioreactor. Blessing asked, how many times can we reuse enzymes? It depends on how they're looked after. All right, but they're catalysts, so they should be able to be reused many times. Okay, so the enzyme can be removed from the bioreactor. That means now, everybody, look, if this enzyme, because it was trapped in a gel, so imagine like this, the enzyme was trapped in the gel, okay, because that enzyme was trapped in the gel, you could take it out. Now you're left with the product. The product now is more pure. Now, great news, because you could take the enzyme out and you could separate it from the product, that means the enzyme can be reused because enzymes are catalysts. If you can get the enzyme and the product separate, because remember, the enzyme turns the substrate into the product. You see that there? Now, so at the end, you got the enzyme and the product together. So if you can separate the enzyme from the product, then you can reuse the enzyme afterwards because they're catalysts. I wouldn't try to stay away from the last one. That makes the process cheaper if you can reuse the enzymes all the time. It's just once or twice I didn't see it in certain marketing schemes. So try to stick with the first three unless you're really struggling. Stick and then use the fourth. Probably two are most likely to be asked. All right, ask for two. Okay, so that's why we immobilize an enzyme, everybody. How do we immobilize it? Attach the enzymes to each other or trap the enzymes in a gel. And the one that we're going to study in the experiment this is the experiment that we're going to be looking at in a minute. We're going to trap the enzyme in a gel, the gel called sodium alginate. Okay? Now, before we go and do the experiment, you need to know one example of an immobilized enzyme that you might use in industry. I've given you two examples. I like this one because we use it in the experiment as well. So, sometimes we immobilize sucrase. So look, we trap sucrase in a gel like this. Okay? And then you put the enzyme, sucrase, into this bioreactor. Okay, that's grand. And then you put the substrate, sucrose. Sucrose is like sugar you put in your tea. Okay, so then you put the sucrose into the bioreactor as well, the substrate. Okay, brilliant. All right, that's excellent. And then the product that's formed is glucose and fructose. So glucose and fructose will now form in the bioreactor. So the enzyme turns the substrate into the product. Now, why would you want to do that? Why would industry want to make glucose and fructose? Well, we often use glucose and fructose as they taste very sweet. They make low-calorie foods taste sweeter. Okay? So you need to know that. Now, before we look at the experiment on that, let's look at the questions they can ask on immobilization. So I want you to go to question 59 or page 59 and look at question 88. And you'll probably notice, if anybody's new to this, all the questions usually follow the order of the chapter as well. And these are all Leaving Cert questions from higher level papers, from mock papers, Leaving Cert papers, and some other questions I've added as well. Now, if you then look at this, what is an immobilized enzyme? The definition: enzymes that are attached to each other or to an inert material. Name two substances that are used to immobilize an enzyme. Sodium alginate is the gel. Now, we haven't talked about that one yet, calcium chloride, because that ties in with our experiment. It hardens the gel. We'll see that a little bit later. Okay, let's look at this here. Give me three advantages of using an immobilized enzyme. The enzyme can be removed from, you got to say from where? From the bioreactor. The product is more pure, or the enzyme can be reused. Brilliant. Now, if you then look at this here, question 94. With reference to any one named procedure involving an immobilized enzyme, state the enzyme, the substrate, and the product. Now, what I often see here is this. A lot of people write enzyme, substrate, and product without saying which one is which. So you got to say which one is which. And if you did, that got you 9 marks, 2.5% of your paper. Okay? Now, I skipped question 91. Make sure you know these definitions. A bioreactor, it's a container in which chemical reactions occur to make a product. To make a product. For those that are members of the platform, you have all these solutions there already. So make sure you have that open. For those that aren't members, don't forget you have access to these questions from this chapter on the question bank. Okay. Now, if you then look at this, everybody, let me go back now, and we're going to go and look at the experiment. Let's go look at the experiment, everybody. We've got 16 minutes left, loads of time. So this experiment is split into 2 parts. So let's look at the first part. And just remember, one last time, this experiment is about an immobilized enzyme. Why do you immobilize an enzyme? And this is why people find this experiment hard, because they don't understand why we immobilize enzymes. We immobilize an enzyme. We trap an enzyme. Sorry, now. Here we go. So we trap an enzyme in a gel like this. Why? Because then when you put the enzyme in here, there's the enzymes trapped in the gel. You just take out the gel. And by taking out the gel, you're taking out the enzyme. Okay? Brilliant. So, let's have a look at this. The first part of this experiment is to prepare an immobilized enzyme. Now, what does that mean in simple English? Trap the enzyme in a gel. Now, what enzyme are we going to use? Well, let's go look at the diagram. So, on this, and you need to be able to draw this diagram. It's a very popular diagram, this one, okay? So, the gel is the reason why you use sodium alginate. Does anybody know why we use yeast? Because when I teach this to revision course students or students that have first time I've seen, most don't know why we use yeast. Why do we use yeast in this experiment? Is yeast an enzyme? No, it's not. It's a living thing. Yeast belongs to a kingdom called fungi. Okay? Yeast belongs to a kingdom called fungi. So why are we using yeast in this experiment? And why to do this? Precious says, can you name both products in the exam? Yeah, I would name both products, absolutely. So well done. We're using yeast because it contains, let me just rub this out here so we can see it again. We're using yeast because yeast contains the enzyme sucrase. Yeast contains the enzyme sucrase. Now, let me be clear in this. Yeast is a single-celled organism. It's just one cell. But in that one cell, there's an enzyme called sucrase in there. Just like we use enzymes in our cells for chemical reactions to occur, yeast do the same thing. Yeast has an enzyme inside that single cell called sucrase. So if you trap yeast in the gel, therefore you're trapping the enzyme sucrase in the gel as well, because the sucrase is found in the yeast. So trap the yeast in the gel, and you're therefore trapping the enzyme in the gel. Okay? So I want you to note there, I want you to go back up here then, I want you to note there, yeast contains the enzyme sucrase. A yeast is a fungus. It's a living thing. All right? So, if we then look at this here. So, I want you to go straight to point number 2. Let's look straight at point number 2. Here we go. It said, and well done for all the answers there, everybody. That was brilliant. I see 1 or 2 people saying sucrose. Don't get mixed up with sucrose and sucrase. Remember, enzymes always end in the letters ASE. Okay? Now, if you look at this, everybody, we're going to mix sodium alginate, the gel, and the yeast containing the enzyme sucrase together. Now that usually gets you 6 marks. Okay? Because by mixing the two together, look what you've just done. By mixing the yeast containing the enzyme sucrase with the gel, you've just immobilized the yeast, and therefore you've just immobilized sucrase, because sucrase is found inside that yeast cell. So that's why you add them to the gel, because you've just immobilized them. Now, here's the thing. When that gel, look at this now, everybody. When that gel comes, when you squeeze that gel out slowly, that gel is soft and gooey. That's not going to keep an enzyme immobilized, trapped for very long. So when the gel containing the enzyme, that we call them beads now, you see there, we refer to them as beads. When you add that to the calcium chloride, that calcium chloride now hardens the gel. So I want you to note there, and by harden, look up here, everybody. Look here. So look at the screen. So there's the gel, and the little yellow or red dots are the enzymes trapped in the gel. See that there? See that there? So that there is the enzyme trapped in the gel. We want that gel to harden, so the enzymes remain trapped. So therefore, we added the calcium chloride. So I want you to note here, okay? Using a syringe, really important you state that. Add slowly, really important you state that. You add your sodium alginate, which is your gel, your yeast, because it contains the enzyme sucrase, we said that up here, okay, to the calcium chloride. For how long? 10 to 15 minutes. Why? To harden the beads. The beads are the gel, everybody. So if you don't know what the beads are, you can just say sucrase, that's the enzyme, trapped in gel. So you're hardening that gel. You're hardening the beads so they remain trapped. Okay? Brilliant. Now, what do you do then? So look at this now, everybody. I'm going to just put a few dots here on this one here. I want to zoom in here. Do you see this one here, everybody? See that one there that I just drew on? See, I drew the enzymes as red dots on the outside. They're stuck to the outside of the gel. Do you think they're properly immobilized, everybody? Do you think them enzymes are trapped? Now, okay, these are, but see this one here on the left, and I've drawn red dots around it. Are those ends, yes, they're stuck to the gel, but are they properly trapped? Are they trapped in the gel? Yes or no? Are they trapped in the gel? Yes or no? Well done. No is the answer. Because, now, we don't want to put that gel with those enzymes stuck on the outside into our bioreactor. Because if you put that into the bioreactor, those enzymes might fall off in the bioreactor. And then you're not going to be able to remove them from the bioreactor. And the product then won't be pure anymore because the enzyme's still in there. So the point is, and this is always a good one to get marks here, rinse the beads, the mobilized beads with water. That removes any enzymes that aren't trapped. That removes any enzymes that are not immobilized. Okay, so if we then look at this here, okay, so there we go. That's 1, 2, 3, 4 key points. That's how you immobilize an enzyme, everybody. That's the first part. Okay, and there's the diagram you draw. So I'm going to show you a question now on this. Okay, so if you look at this, everybody, look at the first part now, and then we're going to finish off with the second part of the experiment then. Now, if you look at this here, all right, sorry, that's the wrong part. We'll go back here. Page 58. To prepare an immobilized enzyme, name the enzyme or the cell you immobilized. So the enzyme is sucrase. The cell is yeast. Make sure you know which one is which, just in case they only ask you to name the enzyme. If they ask you to name the enzyme and you say yeast, you get 0 marks. You trap the yeast in the gel, and by trapping the yeast, you trap the enzyme. Okay? Now, why was sodium alginate used? Because it traps the enzyme in the gel. Why was calcium chloride used? Because it hardened the gel. It hardened the beads. I'm going to be honest with you. This is the question they nearly always ask. This is how they usually ask it. Okay? Describe how you immobilize the enzyme. Describe how you immobilize the enzyme. So remember the 4 points I told you. All right? This is what you say. You mix the sodium alginate and the yeast together. Then using a syringe, now they're immobilized. The enzyme's now trapped in the gel. Okay? Using a syringe, you add slowly. You add it to calcium chloride. Why? For 10 to 15 minutes to harden the beads. Then you rinse the beads with water to remove any enzymes that aren't trapped properly. And that's what, you say that, you've nailed it. So, and people say, oh, this is a difficult experiment. It's not that difficult when you see what you have to write. And it's not that difficult if you understand it. So hopefully now you understand the first part. You immobilized an enzyme. So look what we've just done there. You've immobilized an enzyme. You've trapped it in a gel. So look what we've just done, everybody. Look what we've just done. This is what we've done. You've trapped the enzyme in the gel called sodium alginate. That's it. And that's your bead, everybody. That's your bead that's coming out. That's your bead that's coming out of the syringe. And it's going into the calcium chloride to harden it. Okay, that's the first part. Let's look at the second part. Let's go to the second part, everybody. So the second part of this experiment is examine the application of an immobilized enzyme. So they're basically asking you now, does the enzyme still work even though it's trapped in a gel? The answer is yes. Yes, okay, fair enough. The enzyme is trapped in the gel now, but it still works. It's not only going to work, it's going to make a product that's pure, because the enzyme and the product at the end aren't going to be together. All right, now it's going to be a little bit slower. The reaction is not going to react, there's not going to be as quick a reaction because it's trapped in the gel. But the most important thing is the product. The outcome is going to be more pure. And also more importantly, you want to be able to reuse the enzyme. So yes, it's a slower reaction because it's trapped in the gel, but the reaction is going to be fast. You're going to be able to reuse the enzyme afterwards, and the product's going to be more pure. Okay, so let's look at the second part. So do you see now, everybody, look, do you see these beads that we've made? We're going to use them now. So look here. These are the beads. We're on now page 130, okay? These are the beads. This is the mobilized sucrase. These are the ones we made earlier. The sucrose is, of course, the substrate. The product, of course, is glucose and fructose. So here we go, everybody. We're now testing it. Does this enzyme still work even though it's trapped in the gel? Okay, so let's have a look here. So what we're going to do, everybody, we're going to add the mobilized sucrase into a funnel. They're the beads that we made earlier. Okay, we're going to add them to the funnel. So here, there they are. We're adding them to the funnel. Okay, what else are we adding to that funnel? We're also adding sucrose, the substrate. So that's the substrate here. So now you've got the beads, which is your immobilized enzymes trapped in the gel, sucrase trapped in the gel, all right? And you've got sucrose, the substrate. So look here, everybody. The sucrase that's trapped in the gel, okay, is now going to react with the sucrose, the substrate, because you added them two together in the funnel to produce the product. So look at this here now, everybody. Look, the product now comes out the bottom. And the product is glucose and fructose. So look, how are we going to prove that it's glucose coming out? We're going to use a glucose test strip. Or you could use a clinic strip. You might even use Benedict's solution, whichever you want. But look here, you test the product for glucose. You got to prove that it's glucose coming out. Because look, there's the enzyme, there's the substrate, there's the product. So you're going to test the product for glucose using glucose test strips. So that's why you're using glucose test strips to prove that it's glucose every minute. And there's the experiment. Okay, there's the experiment. Now, you can also refer to the control. Here's the control. I skipped point number 2. Let's go back to point number 2. You're also going to have a control. You're going to add yeast that was not immobilized. So yeast that was not in a gel. Just normal yeast. All right? That contains sucrase, of course. And you're going to add that to another funnel. So look, you've got free yeast, which contains the enzyme sucrase. Okay? So look, the same enzyme, the same substrate, the same product. But this enzyme, and therefore that yeast, was not immobilized. So that's the control. So you're going to get, it's the same enzyme from yeast that was not trapped in a gel, the same substrate, and the same product. And I'm going to ask you now this, everybody. Can you tell me which product we're going to call, because it's the same enzyme, the same substrate, the same product in both. It's just this enzyme is immobilized in a gel. So can I ask you, product number A, glucose and fructose. Product number B, glucose and fructose. Same enzyme, same substrate. This enzyme is immobilized. This one was not, though. Which one is going to be more? Which one is going to be the pure product? Is it glucose and fructose from A, or is it glucose and fructose from B? Which one's going to be more pure? Which one's going to be the pure product? All right. Usually, are these experiments connected, or would they be two different? No, it's the one experiment. It's the one experiment. It's just two different parts. It's A, everybody. Well done. Why is A the pure product? Because I'll tell you the reason why. Because this enzyme is trapped in a gel. All right, so look here. Let me show you this here. Do you see the beads? Let me just see here. So there's the beads, and there's the enzymes trapped in the gel. See there? Those beads cannot get through the funnel. So the enzymes are trapped in the gel. They can't get through the funnel. Only the product comes out the bottom. Over here, this enzyme is not trapped in a gel. So it just passes through with the product. And this product's not pure. So, but generally, when they ask you to describe this, you just describe the 4 points. I'd always include the control. If they ask you about the results are the immobilized sucrase, that's the beads you made, all right? That produces a product that's pure, that's clear, because the enzyme wasn't in it. The enzymes were trapped in the gel, which were still in the funnel above. But the control, which is the enzymes that weren't immobilized, just normal yeast, it produces a product that's cloudy, that's not pure. Why? Because those enzymes passed through the funnel because they weren't trapped in the gel. So let me show you final time today. All right, let me show you this here. Page 59. Examine the application of the immobilized enzyme. So make sure you don't get the two parts mixed up. This is examining the application. The first part is asking you to prepare to trap it in a gel. Okay, but this time you've trapped it in a gel. Examine its application. Does it work? So here we go. Okay, so it says, describe how you examine the application of an immobilized enzyme. So what we do is, you add immobilized sucrase into one funnel, and then you add sucrose to that same funnel, the substrate, and state that it is the substrate. Then the product comes out the bottom, and you got to prove that the product is glucose. You're going to use a glucose test strip to do that, and mention it every minute, because we want to see which one's the fastest. All right, the immobilized sucrase is actually slower because it's trapped in the gel, but it's the product's more pure though from it. The results are, I would always state the results. Oh, sorry, and always reference the control. The control was you added yeast into a separate funnel, and that was the yeast with the enzyme that was not immobilized. So the results are, always reference the results. The immobilized sucrase produces a product that's clear. All right, the free sucrase, the yeast that was not immobilized, it produced a product that was cloudy. It wasn't pure. Why? Because the enzyme passed through the funnel as well. Now, my advice to you is, please make sure you know which diagram is associated with which part. This diagram is how you immobilize the enzyme, how you trapped it in the gel. This diagram is how you examine its application. Does it still work? Now, that's it, everybody. I'll take a few questions now, but that chapter has been usually worth 7.5, sometimes up to 13-14% of your paper. Hopefully it's like that again this year. So look, what are we going to do? And this is the timetables. I'll put the timetable up in the news feed again as well. But let me just show you, for those that are on the platform and have access to my H1, oh, that's the wrong one there. So for those who have access to my H1 biology platform, all right, so you see it there. So you look here, you'll see the timetable there on the very right. And you'll see, there we are, everybody. That's what we've done today. Tomorrow, 7:30 to 9:30 respiration, massive one. Then the next day is an easy one again, cell division. And then photosynthesis the day after, big one again. And then the last one's a big one. So 30-40% of your paper, everybody. Now, my advice to you is, you know what you should do now, all right? All right, go through the rest of the questions in that question bank. Go through the solutions. For those that have access to that, you'll see the solutions on the platform. But even those that are here for free today, go through you've got the questions in the question bank from that chapter. Go through them now and find the questions you don't know in my notes, and you'll see them there. So look, everybody, brilliant class today. I hope you find a great interaction. It was 2 hours on the dot, and we did it from start to finish. We left nothing out. Hopefully you feel now you understand it. So I'll see you tomorrow at 7:30. For those that have any questions, I'm going to be around for another few minutes, so ask them. But if you're happy enough with that, well done. Go look at it again. And I will say this to you. Look this. All right, because I know some people are going to be asked questions about this. All right, Unit 1. Sorry now. Oh, here we go. So just take a wee second. There you go. Unit 1 is... Sorry, now it's jumping there. There we go. So Unit 1 will be 25 to 32.5% of your paper. Those 4 chapters from Unit 1. The 7 chapters that we're studying this week are 30 to 40% of your paper. Okay? These 4 chapters here in bacteria should... Now, I'm not saying these are going to be bang on accurate. That's what's happened the last 3 or 4 years. 10 to 20%. Thank you so much. If everybody is happy and they want to head off, thank you so much. Thank you so much. But if anybody's got any questions, ask away. The human biology is 30 to 45% usually. All right. Genetics is 15 to 30% usually. And the plants is 20 to 35 usually. Obviously, that does not up to 100 because you have to do... You have loads of options on the paper. You don't do them all, of course. Okay, so let's see some questions here. Cal, is it possible to completely leave out plants reproduction? Yeah, you can leave out plant reproduction. It comes up nearly every year, though. Thank you so much, everybody. Comes up nearly every year, but yes, you can leave it out if you want. If you don't like it, it takes up too much of your time because you have so many options in the paper. The only thing I'll be careful of, there's 2 experiments in plant reproduction. So thank you so much, everybody. If you ask questions, there's a lot of thank yous there, so really appreciate it. But if you ask the question, you might type it out again because there's Lots of thank yous there. So you might just type it out again so I see the question at the bottom. But yes, Carl, the answer is yes. You can avoid plant reproduction. Absolutely. And still be able to do a full paper. Let me look through here. Would you be able to make a video on genetic engineering? Faye, I will indeed. That's another little short one. I'm glad you're using that on demand. I think 95% of the chapters are there. There's a couple I have to tidy up on it, okay? Lucy, will free members be able to catch up on a day of the grinds this week if you miss it? Lucy, I don't think so, Lucy. You don't have access to the platform, all right? Caitlin, I have a question. So, apologies. So it's coming up the last few weeks of school, and I'm thinking about taking time off school to revise. Would this be a smart plan, or would you be better to continue going into school? It depends on a lot of factors. It depends on your teacher. It depends on you. And I know sometimes I always cover so much in class, and up to the very last minute of the very last day, I'm doing exam questions, go back over material. But then, even though I do that, I know one or two of my students would prefer to stay at home, and they do that. So it depends how dedicated, how strict you can be at home. So that's different to every individual. But if you think you can be really strict at home with yourself and do hours and hours of study, and you know the course, then maybe, yeah. But if you're on the other hand and you pick up one or two little things, then it might be better to be in class. It's hard. It just depends on the individual. This class will be uploaded the next, hopefully tomorrow, I'd say. It says 48 hours, but it's usually tomorrow. Let me see, any other question? So which diagrams do you draw if you're asked about experiment as a whole? Eve, that last experiment, they usually ask you about both parts. And if they ask you to draw a diagram, they specify which part. So if they ask you to draw the diagram how you prepare the mobilized enzyme, that's the syringe and you're trapping it in a gel. If they're asking you to describe how you examine its application, then that's the second part. All right, so you be confident in which part they're referring to, and then you're drawing the right diagram. Kieran says, Wesley, there's a lot of questions coming here. Hold on, Kieran, what are you saying here? So, Kieran says, what's the best way to retain this info? Since tomorrow we're straight into a new class and I won't find a time between. Kieran, the best way, I would say now, Kieran, go through as many exam questions from that question bank. Because when you know the questions, then you're really confident. And Kieran, 3 of them experiments are the exact same. So you know 1, you know 3 of them. So there's a lot of good things in that chapter. Emily, would I be better leave out plants or DNA? Or what's better? Look, plants are usually worth more than genetics, but not always, but usually. But I can't answer that for you, Eve. I can't answer that. To be honest, Eve, I'm going to be honest with you. If you had some of plants studied and you had some of genetics studied, then you might be okay as well. But look, I would love to say to you, if you left out the 4 genetic chapters and you left out the 5 chapters on plants, you'll definitely be okay. There is a slim chance you might be, but I wouldn't take that risk. So I would definitely have one of those sections studied. These are the 2 common things, genetics and plants. That's what I keep hearing. If you're very, very lucky on the day, you might get away with it, leaving both out. But that's I would say, the chances of that's a high risk. So I would try to have some of them studied, at least in some part, okay? Now, that's no problem, Clodagh. Is it possible to go from H6 to H2? David, as I said, 350 students I taught last year in the day school that I used to teach in. 61 or 60% of them go to H1. Out of that 61%, I would say, so that's over nearly 200 H1s out of the 350, whatever it was. I would say about half of them are getting H4s, H5s, and H6s in their mock. So you put the work in now, David, and you know those questions from the question bank and so on. You know the chapters really well that you studied. Then you'll get it. You will get it. All right, just get going. Any other questions here? A lot of questions. Anna, just wondering, how do we receive our mock grade? Anna, the people that did the mock at Easter in this Talbot Hotel that I gave them, it'll be emailed out to you. I'll hopefully get them out this week. Sarah, what chapters would I need to get a H2? Sarah, that's such a broad question. Sarah, all I'm saying is this. Study the sections. Unit 1, 25 to 32.5%. Unit 2, those 7 chapters, 30 to 40%. The first 4 chapters in Unit 3, bacteria down to viruses, there's 10 to 20%. I honestly think, if you got that started off, those 4, those 7, and those 4, my goodness, you'll have a big chunk of your paper already studied. and you'll be full of confidence, and then you'll hopefully then go into human biology maybe. Amy, got H3 in the mocks, and I want a H1 in the real thing. Is it possible to make a comeback? Oh my God, Amy, of course it is. A H3 is, you're so... Oh, Amy, I tell you, I got students H5s, H6s in mocks and got H1s. So, absolutely, be confident in yourself. All right, and remember, there's so many options in the paper. So, there's questions flying in here, so I'm trying to keep up. Are you going to do a class the week of the Leaving Cert? So, I'm doing these 3 biology blitz classes. Or weeks. And then I'm doing every other week in between. The week before your Leaving Cert, I'll probably be doing classes every day. The night before your Leaving Cert, I'll be doing classes for those who want to be there as well. So I'll be there the whole way up to the very end. Lily says, what human biology topics should I prioritize? To be honest, human biology, last year, the majority of human biology chapters came up. Last year, human biology was massive, over 45% of your paper. So look, it depends on the year, but like I always say, the big 5 that I'm doing in my biology blitz, the big 5 that I'm doing my biology blitz in the May the 10th, the heart, human digestion, excretion, the nervous system, human reproduction. I always say they're the big 5. You'd be unlucky if 2 or 3 of them didn't come up. All right? Then the other smaller chapters, it's hit and miss. It really is. So know those big 5, and then like the smaller chapters then are sometimes easier too. So look, it's very hard to say because Lydia, this is the thing. I used to always predict papers inside out, and I predict the most of last year's paper too. But the problem I'll say to you is this: the majority of the course is coming up because of the extra options on the paper. So that's what I'd say is over 2/3 of the chapters will be asked. All right. So and a lot of those are the big ones as well. The big chapters. Any other question? Hold on, I'm going back over some of these questions. Amy says, can I leave out of plants and plant experiments because I'm only answering unit 1 and 2 experiments? The answer to that is if you know your unit 1 and 2 experiments, so question 8 and 9. But I will say, Amy, the experiments in question 10 can come up anywhere as well. Experiments can come up anywhere in the paper. But it's you'd be unlucky if they did, though. Let's put it that way, Amy. Okay? Emily, could I leave out human biology apart from blood and the heart? Or would it be... Oh, Amy, you're leaving out a lot. If you have everything else studied really well in terms of the entire course, genetics and plants, and you leave out human biology, like, you might be all right. There's not many experiments in human biology, so section A, your options would be definitely limited big time. And you'll be relying on all the other sections, and some of them other questions can be difficult. So definitely have as much human biology studied as you can. Precious, I've struggled with the lymphatic system. It hasn't come up in a long time properly as a question. I don't know if you're on the platform, Lydia, but if you are, go watch my on-demand on the platform. You'll understand the lymphatic system then really well because there's only 2 short videos, and I do it inside out. Let me go back here. Am I leaving out? Okay, I'm leaving out fungi. Fungi was a big one last year, actually. I think fungi, from the top of my head, fungi was 60 marks, 15% of your paper. So, look, I do think something, it's very, you're probably less likely to see fungi this year, but it still can appear. There's so many chapters will appear because of the size of the paper and all the extra questions. But yes, if you leave fungi out, obviously lots of the other parts of the course, you'll still be all right, of course. All right. So if you're stressing about 1 or 2 individual chapters, then you're going to be okay for 1 or 2 individual chapters. But like if you're leaving whole sections out and you're leaving more than one section out, the way I split it up there, then you're taking risks, of course. All right. Look, let me see here. Can you give me a general idea of timing and how long we need to spend in each section? So look, timing's not an issue, but usually, because most people finish biology for 40 minutes to spare, or half an hour, but usually, most students spend about 25 minutes on section A. Most students spend about 20 minutes then on your 2 experiment questions there. So you're talking about 45 minutes there. So you still have 2 hours then, all right? 2 hours. So most people then, you do your 4 questions, they're supposed to be half an hour each. And so that's supposed to take you 2 hours. No way will that take you 2 hours to do 4 questions there. Time's not an issue. Don't be worried about time biology, all right? Unless you're going really slow and reading questions like a snail. And I appreciate sometimes people do need readers and so on. That's fine. You get extra time. But what I'm saying is time's not going to be an issue for most people. All right. All right. I'm going to go back and look at a few more questions before we leave it here. Because there's hundreds of questions. Let me see here. You see, I think I got a lot of them answered. Daniel, my 3 questions are, I got a H6 in my mocks. I answered that one. Is it possible to improve from a H6? Absolutely. And what else did you say, Daniel? If I leave out genetics, and what is the best way to study and improve my... The best way to study, Daniel, is study my notes on that chapter, and when you think you know the chapter, then go to my question bank and do all the questions in the question bank on that chapter. Answer the ones you find. Look at the ones you don't know and figure out what they are. If you're, I don't know if you're on the platform, Daniel. If you are, then have my question bank solutions opened up, so you're not wasting time. Then you'll see the answer straight like that. Okay, that's what I would do. Okay, I'm looking back through a few more questions. Will any of the immobilized enzymes fall into the beaker, or is it no? Sean, the immobilized enzymes are trapped in the funnel. And even if they did fall into the beaker, you could pick them out because they're gel. You just take out the beads. You just take out the gel and you're taking out the enzyme. Okay? But no, they're trapped in the funnel because they're too big to pass through. Sean, will any of the immobilized... No, I answered that one. Sorry, Sean. Let me see here. Can you give me a general idea on time? No, I did that one. From this point, would you recommend just doing exam questions? Kian, it depends on how your knowledge is. If you have the knowledge and you know the knowledge, then exam questions are key. That would be good advice. But like, make sure you know your definitions. Keep looking over those definitions all the time. Do not take the definitions for granted. It's often the one that... Definitions are often the thing that makes the difference between H1 and H2. Because people might understand the course and not get a H1 because they didn't know the definitions well enough. You might understand the chapter, but that doesn't mean you know the definitions word for word. So I would just make sure you still know things like definitions and practice them. Okay, let me go down to the bottom here again. Any other questions flying through here? I find the plant section difficult. But I want to H1. Do you know of any advice? Look, I would just say you could leave out plants. It could be a big part of the paper this year, but you'll still be able to do a full paper without them. The only thing I would say is, don't rule out all the plants. If you're on my platform, pick 2. Look at even do the first 2 the chapters. I love the first 2. They're lovely. So if you're on the platform or you have my notes or just it's your own school, try to know the 2 first plant chapters anyway. And at least then you're not ruling out all the plants and you'll be able to do some plant questions. Okay, I think we're nearly out of time. And I think I've answered a lot of the questions. Could you make a list of chapters that include having to give an account of certain things? Could you make where did that question go? Could you make a list of chapters that include having to give account of certain things? That doesn't make any sense, Olivia. Every chapter kind of has stuff like that. So every chapter has where you have to describe a kind of things. Like, so I don't know what you're asking there. Lengthy. Are you talking about lengthy answers? Like a lot of chapters, Olivia, like cohesion tension model in plants, the development of female gamete plant reproduction. Olivia, my advice is just Olivia, get studying the chapters, and go into my question bank, and look at those questions. And that there's not many of them, but the answer is go and look at my question bank and you'll see the solutions there as well for them. And they're in your notes. So some chapters like the active site theory that we did today, that requires 4 different points. The cohesion tension model for plant transport requires 4 or 5 points. So there is a few of those questions there in the course, but don't worry about them as separate. Just go and learn the chapter and look at my questions on the question bank on those questions, and you'll see very quickly the solutions I've given you on them as well. All right, so I would just focus on the chapters, Olivia, and when you come across those questions from my question bank, you'll see them there then, okay? That's my advice, all right? Okay, everybody, see you tomorrow. How do I get the notes, Sarah? Sarah, I assume you're not on the platform, are you, Sarah? Are you on the platform, Sarah? Like, are you a member or are you here just for free today, for this week? So this here is in my H1. It's on the first page of my question bank, and it's also on the first page of my actual notes. So look, here's the question bank. Hold on now. So look, if you go to, oh jeepers, hold on now. Where did that go? So look, there's the question bank. So look, it's all on the first page there. See it? But I'll put those 3 pages on the news feed as well. I'll put those 3 pages on the news feed. Zara, because you're not a member, you won't get access to obviously the entire digital copy of the entire notes. But if you go to the news feed, Sarah, you'll get today's enzyme notes. Look there, Sarah. You go to the news feed. There's the enzyme notes there. See them there? That's the ones we use today. So I'll put up the notes we use every day on the news feed. And there's the questions on the question bank from today as well. So you'll see them there as well. But in terms of the entire course, unfortunately, I can't, Sarah, give you access to them, unfortunately. They're for the members only. All right, everybody. I'm going to go have a cup of tea and maybe get a fresh air. So I hope you find that good. Of course, a few times from now. When I study a chapter, I feel like I forgot it. And Emily, that's where it goes. Go do the questions. Because then once you become so familiar with the questions, so study the notes and then do all the questions and keep looking over those questions and do the questions you find difficult. Thank you so much, everybody. Thank you so much. All right. If I didn't answer your question today, ask me it tomorrow again because we've got all week. So, blessing. But you are a very good teacher, but do you teach also chemistry? It would be really helpful. Unfortunately, I'm only a biology, but there's great chemistry teachers on the platform too. So, but I don't know if the chemistry teachers are doing this, but anyway, I shall leave yous and I will talk to yous tomorrow. Tomorrow's a big one, everybody. Tomorrow is respiration. All right. Blessing. Thank you for that lovely comment. I really appreciate that. Clodagh, I'm leaving out genetics, but covering everything else. Should I just leave out genetic crosses, or what do you? So, look, I will say one thing about that before I leave. Where is it here? So, Clodagh, if you're leaving out the genetic chapters, don't leave out the DNA experiment though, because the DNA experiment is the last experiment on question 8. So, yes, if you leave out the 4 genetic chapters, you'll still be able to do a full paper. But look, don't rule everything out. Like, even have some of the chapters or parts of the chapters studied so you have an idea, all right? But make sure you have that DNA experiment studies if you do decide to do that. All right, everybody. Daniel, is it free? I'm not on the platform. Is what free, Daniel? Is what free? Those enzyme notes are on the platform, Daniel. And the questions from today's question bank are on the platform. You can download them as a PDF. Okay? Is that all right? I think that's what you're saying. But everything else, unfortunately, all the other biology blitz, the biology, I'll just show you I better show you this one last time. So look, there's the timetable. I'll show you the timetable one more time, and then we're out of here. So look, the first week's free, but the other 2 weeks, they're only for premium members. I do know they're doing a great offer at the minute. I think they're doing a great offer for the last 6 weeks. So if you do want to, no, it's still money. I can't quote. Admin might confirm that, but is it €190 or something? I might be wrong. Admin might confirm that on me. But if you want to come along to those last 2 weeks, or if you want to get, I'm going to be teaching every week, and 2 of those weeks I'm going to be doing another 10 hours and 10 hours over those 2 weeks. But even in between that, I'm doing Sundays, Tuesdays, and Thursdays all the other weeks as well. So you'll get a lot of benefit if you do want to join for the last few weeks. But if not, come along for the next 4 weeks, or 4 classes anyway, over the next 4 days. All right, everybody. So Daniel, any advice on making my own then? On making what, Daniel? What do you, the study plan. Sorry, Daniel. I do have a study plan, Daniel. I do have a study plan. I'll show it to you here. It's on the platform for the premium members, but I'll show it to you here as well. Here it is there. There's the study plan. So, Daniel, the first 3 weeks of that study plan is we've already studied, unfortunately started, it was a week ago, but the first 3 weeks is 55 to 70% of your paper. Now, the good news is, the second week, cell structure, cell diversity, cell division, enzymes, and the third week, respiration, photosynthesis, diffusion, osmosis. They're all the chapters we're doing in the next 5 days. All right. And then you study unit 1 on top of that. You study 55 to 70% of your paper. That's how much. Look, I'll finish off on this point. That's how much. And there. So look, I'll put that up in the news feed as well. I'll put that up in the news feed during the week as well for you. But that's how much the first 11 chapters, these 4 chapters and these 7 chapters are 55 to 70% of your paper. And last year was 69%. So you can cover you can cover 11 chapters, them first 11 chapters, and you'll have 55 to 70% of your paper revised. All right, everybody, I'm going to see you. Have a lovely day. I'll see you tomorrow at 7:30. Thank you so much.